1953. This article came in which journal? कौन से जर्नल में पब्लिश हुआ था? Okay. Then after DNA structure, we had something like discovery of restriction enzymes by Missels and Stahl. This was around 68 or so. And then in 1972, there was production of recombinant DNA by Cohen and Boyer. And it was in 1983 when there was demonstration of agrobacterium-mediated transformation in plants, like this is Estrella et al. There were in fact a series of papers, I would be talking about them. And then in 1994, it was the development of flavor saver transgenic tomato by Calgene, company by the name of Calgene. This was one of the first commercial product and very important. So all these discoveries and the DNA being the underlying molecule, it has brought in all living forms into a single gene pool. That is very important, the underlying principle. So transgenic technology, it provides enormous power, enormous opportunities, but it comes with enormous responsibilities too. We are tinkering with the nature. We have to be sure that whatever we are doing is right. Like, I won't be essentially following it, but to put it in perspective, it is basically, we'd be talking about the DNA, you locate the gene for the plant traits with the breeding. We can't do away with the genetics. Biotechnology can't be practiced all alone. It would rely on several streams, several disciplines. That is what biotechnology is about. Some of you working on transgenics during your PhD, all of you are PhD students, right? Most of them. MSCs. So what are you working on? समय ज़्यादा नहीं है, चलिए आगे बढ़ते हैं. So like some of the tools which are very important, I would say the restriction enzymes, they are very, very important if you are making the transgenic plants. The ability to is very, very important. And another important in very simple ways. There are types of ligase depending upon the which particular molecules you want to join. So lot of scope is now with the structural genomics really established very well. We can do the genome sequencing very quickly. This was not so true about 3 decades back. Okay, so now you can unravel the genome sequence of any organism. That is not a big deal. But still, locating the gene for plant traits or identifying the gene for plant trait, that is still a big question. Okay, Arabidopsis or 16 GB genome of wheat, that does not really make much sense. It would be 80 GC. But what are the genes specifying which particular trait? That is very, very important. Okay, so important trait. It is one of the most difficult or limiting step in the transgenic process. Relatively little is known about the specific potential, improved stress tolerance, be it biotic stress. Now we are much more conversant, be knowing about, say, about 50 to 100 genes. All of you would be familiar with it. Agrobacterium as a vector, KB odd. So you provide the vir function in trans, दूसरे molecule के ऊपर. And basically you are combining the 2 vectors in each agrobacterium cell. You have something called as the plasmid. उसमें tumor production के genes नहीं रहेंगे. FG is your favorite gene. And you have a MCS. What is MCS? Multiple cloning sites हमें क्यों चाहिए होंगी? क्या ज़रूरत है? जो हम इंसर्ट करना चाह रहे हैं डीएनए फ्रेगमेंट या जीन, तो उसको क्लोन करना पड़ेगा ना, किसी एंजाइम से उसको कट करेंगे और एग्रोबैक्टीरियम के डीएनए को भी कट करेंगे, कंपैटिबल एंड्स बनाएंगे एंड इवेंचुअली इट विल बी इंटीग्रेटेड. और जितने एंजाइम्स होंगे ज़्यादा होंगे तो उतने बेटर होगा. दैट विल गिव यू ऑप्शन टू पुट इन मोर जीन्स एंड ऑल. ओके, बट नाउ अगर ट्यूमर हो जाता तो पता लगता है भाई इट इज़ नॉट डिज़ायरेबल. So how can you detect the cell which are carrying the tDNA inserts? it is here the selectable markers come into picture, the role of these markers, okay, because it would be a very small fraction of the host cell population. यहाँ में host cell कौन सा cell हुआ? Bacterial cell, the plant cell, host. So the bacterial cell population, बहुत कम cells actually transform होंगे, okay, and eventually you use the conditions in which transformed cells will live and untransformed cells will die or they will not grow. So basically this all is the selection pressure, and this is something called as the negative selection. There are some positive selection also. We have specific genes like PMI, but the need of the and one of the authority, very big name, okay. You have got usko easily up, you basically call it Gus staining, okay, and there are histochemical methods, there are fluorometric, okay. This was from one of the plant cell article, this level over here, like transient, it has not integrated into the host genome, but this green one is the host genome. If this tDNA it gets integrated over here, this would be the stable transformant, and eventually you regenerate and get the whole transgenic plant, okay. And this was from one of the reviews at Purdue University, and he is the pioneer authority, okay, and he's still very much active. He's a distinguished professor of biology. So like these are the pioneering people. Like this work started somewhere around late 70s or so, okay, and he commented upon, I've taken from this very article, in this bacterium, and the list, and like very beautiful article and gives a lot of things to ponder upon. Milestones in plant transformation. First integration of agrobacterium tDNA in plants, and eventually goes to the world food security. That is essentially what we are looking upon. So in past 40 years or so, it has all shaped up. Just comment upon, okay. So this was a landmark year. These are the pioneering articles on plant transformation. So first one was from Mary Dell Shilton, and I have a photograph over here. She worked at Washington University in St. Louis, and the title was Regeneration of Intact Tobacco Plants Containing Full-Length Copies of Genetically Engineered tDNA and Transmission of tDNA to R1 Progeny. This was April 1983. This was a Cell, the topmost journal in the world as far as the primary research is concerned. Then there was a second article, again in 83, that was a Nature article. Again very respected journal and with impact factor of more than 60. The expression of chimeric genes transferred into plant cells using a TI plasmid derived vector. And this was basically work of Mark van Wontegu and Jeff Schell. They all are from Belgium. They're basically European molecular biologists, very big people. And then the third article, it was again from Mary Dell Shilton. It was a chimeric antibiotic resistance gene as selectable marker for plant cell transformation. It was from Michael Bevan's, like it was, in fact, Michael Bevan is a student. So all this, so 1983, and we are, so past 40 years, it has been very interesting and lot of work has been carried out. So these are the pioneers. And of course, as I told you, the plant breeding is again an integral part of it. Okay, you will check the activity of the introduced gene, you will check the inheritance of the gene, and you have to take care whether there are some undesirable effect. Many times, now of course, we are much more wiser. We can direct the tDNA also, but otherwise primarily we don't know where would our tDNA land up. And the method of integration, it is called as the illegitimate recombination. Okay, so अगर कहीं आपका tDNA कहीं integrate कर गया कोई more बहुत important gene में, तो that would be silenced. That could have a catastrophic effect. So all those things are important. I don't have much time. So, sorry. So this is the biolistic method, and in it you use a gun basically. It is just gene gun, and there is a company by the name of Biorad. Okay, and it is called, they have a machine by the name of PDS, Particle Delivery System, PDS Helium 1000. They use the power of the helium gas, it is pumped through the cylinder and all. Then you have got the basically some inert particles. Some inert particles, usually they prefer gold. Many times tungsten particles are also used. You coat these particles with the DNA and then bombard your tissue, literally. Okay, there are macrocarriers, microcarriers, stoppers and all. Then you see, Many times it gives very good result also. But like it comes in fragment and all, those kind of things. So like if you put it in perspective, you start with the explant, go for agrobacterium transformation, go for particle bombardment, maybe you can produce a calli, you select the unsuitable medium, eventually you have something like putative transformed embryogenic calli, you select the transformed cells, go for the regeneration, and eventually do the basic molecular biology, go for the southern analysis, see whether the gene has integrated into the genome. If you are doing it at a much more advanced level, you can do the Western, you can have the antibodies designed for that particular protein, go for that, like particularly for the cryogenes and all, and eventually you land up with the transgenic plant. This, like, but again, let me tell you, this is at one time when we started a student of biotechnology, they used to say कि बहुत जल्दी हो जाता है ये काम, but no, it also takes a lot of time. Probably by the time you start the experiment, you end up with a real transgenic plant which can be released. Of course, within big inverted commas, it's a long drawn process. It can take 7 to 8 years. There is no shortcut. Okay, but now we talk of speed breeding and all. So some data on transgenic crops. Okay, I would move to the major one. This is what I found from the ISA, ISAAA. This is the latest data. This is 2018. Around, with respect to transgenic, they are being cultivated on an area as large as 190 million hectare. Okay, that is very big. I don't know, India's total area would be somewhere around 300 odd million hectares, the total cultivation. So to put it into perspective, the total worldwide grown area is somewhere around 190 or 200 odd million hectares. And there are a large number of plants which are being used. And biotech crop area has increased 113-fold. This is somewhere around 1996 when it started up. And the major biotech crop, which would be the major biotech crops, सबसे ज़्यादा वर्ल्ड में कौन से ग्रो कर रहे हैं? Soybean is the number one crop. Nearly 50% of the acreage is with respect to soybean, followed by maize, followed by cotton. Okay, these are the major crops, and we talk about 4,349 approvals for so many events. We talk in terms of event, and this is the world map. 70 countries they are adopted this biotech crops. Like some of them they are using, doing both R&D. Some of them they are importing, exporting, and large number of things. So try to appreciate the number of countries, despite so many regulations and all, the technology, it has been embraced by a large number of countries. So it has to be promising. And let me tell you, you and me saying that transgenics are okay really does not mean much. The farmer, जिसका livelihood उस पे dependent है, if he or she is reaping the benefit, then only they will plant. मेरे कहने से कि मैंने लैब में बहुत अच्छा काम करा है, आप लगाइए उसे, they will not do that. Okay, because their livelihood is dependent on it. We are doing it as a research. Okay. So large number of advantages. Few success stories. BT cotton in India, which is the crop which is being approved. BT cotton, almost 95% of the cotton which is growing in India is now BT cotton. Okay, the delta endotoxin, BT toxins, and at our institute also we have got large number of genes, like these are basically cryogenes. So large number of cryogenes, they have been isolated, identified, given to several companies. Our earlier director, Dr. Anand Kumar, he has worked upon it and shared lot of genes with the private companies. Okay, so this has been a big success story. There's something like transgenic papaya. All this is old, but just to comment upon that, this has been commercially cultivated in Hawaii. There was amaranthus gene. This was JNU's work primarily, and you have got improvement in oil. You have got high oleic acid, lauric acid. The golden rice, very important technology, again came from Europe, Ingo Potricus lab. Large number of genes. It has been also, there was a big program in India also. They want to integrate into the different lines. And coming to India, so there is only one crop, that is BT cotton, which is being grown commercially. And just look at the fate of the BT versus non-BT. This is the BT, hale and hearty ball, and this is just shriveled away, gone for good. Okay, and this was again a very fundamental and very important like development with respect to the development of transgenics in country. This is the ball guard development. So it was MICO, like there was an institutional biosafety committee which was established in MICO. What is MICO? Maharashtra Hybrid Seed Company. Okay, and this has been behind it. It was established in 95. Large number of seeds were imported from Monsanto. Back crossing was done. Studies were conducted, and then data was evaluated in 2001, and eventually it was commercialized on 5th April 2002. It has been almost now nearly 22 years since its commercialization. A lot of the new versions have come up, Ball Guard 1, Ball Guard 2 and so on. And coming to BT brinjal, this was again important, and this BT brinjal, it was resistant to this fruit and shoot borer. Again, it was developed by the MICO. There were different events. There was one particular event by the name of EE1, and even our center participated in it, and we had generated a gene by the name of CryFA1. And but on February 9, 2010, Ministry of Environment and Forest, at that time it was MOEF only, it placed a moratorium on that. No more research on transgenics. Okay, no more release. No more release. Research can continue. Okay, way back in 2010. Okay, then the then Environment Minister did that. So, what were the concerns? Okay, so they say India is a mega diverse, one of the 12 mega diversity country. It has large number of recorded species, and so on and so forth. But let me say that there is very good biosafety mechanism in our country, which has been constituted and which is being followed religiously. You have got something like recombinant DNA advisory committee. At the institutional level, you have institutional biosafety committee, RCGM, and the apex body is something called as the GAC. It was earlier called as the Genetic Engineering Approval Committee, but in 2010 it was called as the Genetic Engineering Appraisal Committee. That is one of the apex body. So this is the scheme of thing. Like if you are working on it, you have to submit your applications to IBSC. It goes to RCGM, eventually to GAC. It gives the permission for conducting the trials, BRL1, BRL2. It's a long process. At our institute, we are also working. One of our scientists, she is trying to release the pigeon pea, and it's a long drawn process. Okay, despite us being a government organization, even for us it takes very long to get the required approvals and all. Okay, so the major safety issues they comment upon: biosafety, labeling of GM versus non-GM, antibiotic resistance gene in GM food, environmental concern, gene transfer, effect on biodiversity, public awareness. Probably I would rate this as one of the major bottlenecks, and socio-economic issues. And the decision on GMOs, like you think in terms of food safety, environmental safety, agronomic benefits, transparency. You have to basically do a SWOT analysis kind of thing. What are the risks? What are the benefits? If the benefits are more, go for it. And no technology is 100% risk-free. You just can't say anything. And lot of debate, GM food, yes or no. Primarily, I would say it is driven by ignorance, conservative thinking, and fear of unknown. Pata nahi kya ho jayega. And the 4 E's about the transgenic plants: they are easy, effective, economical, and eco-friendly. And public awareness, it is absolutely necessary, and decisions, they need to be based on scientific facts and available options, and not on exaggerated and unfounded fears, like organizations like Greenpeace, like it has come from the grocery store. There is no need to be panicky about that. Think about it wisely, whether as a country like ours with more than 1.4 billion people to feed, can we really have the luxury of doing without the GM? Okay, and coming to like the theme of this all training is so these are some of the products of the CRISPR technology, and of course we are now reaping the benefits. There was an article in Nature Biotechnology in December 2021. It talked about GABA enriched tomato. It was the first CRISPR edited food to enter the market. What is GABA? GABA amino butyric acid. Okay, and this I just took from the net. And this was the first CRISPR therapy which was approved by FDA, Food and Drug Administration, or USA. And on December 8, 2023, they have approved something called as the Casgevy. It's a groundbreaking CRISPR-based gene editing therapy from one of the Vertex Pharmaceuticals for the sickle cell anemia disease. So things are slowly moving. The technology is there. The products are coming. And so with all this, I commented upon that moratorium on the GMOs and all in 2010. And it was only in March 2022 they talk about giving the permission for the genome edited plants. Okay, and the particular ones like SDN1, SDN2, like we will be talking about them. Site directed nucleases 1, SDN2. Okay, so this is a very welcome step from Government of India, wherein they are saying that they are not regulated. It's SD1 and SDN2. You can release them. Okay, if you have it, go for the release. So this is a very important change, and this is from ISA side. I just took it to show, like, this is the gene editing resource, and this all technologies like CRISPR reveals how much circular RNA undermine rice disease, gene editing boosts resistance of kiwi fruit against canker disease, Chinese researcher improve biotic stress tolerance of cotton. This is from June 26-24 to only January 31. Like, I didn't have much space. So gene editing of grape, it enhances aromatic attributes. CRISPR improves leaf rust resistance in wheat. So many developments are taking place. So many products are coming up. As a country, we just can't afford to stay back. We have to venture into it and really come something. This is where I would conclude. So this is from Norman Borlaug, a Nobel Prize winning agronomist. World peace, it will not be built on empty stomachs and human misery. Deny the farmers access to modern technology and world will be doomed. Not from environmental degradation, as some would have us to believe, but from starvation and social and political chaos. So the food security is very important, and we just can't think that we can do without the technology. The technology is very much here to stay, and it would be better if we embrace it with everything. That's all. Thank you very much. Anil, ज़्यादा time तो नहीं है?
So we would like to thank Dr. Jain for very nicely summarizing the basics of plant transformation and giving you some, dimensions of regulatory framework in India. So thank you, sir, for accepting our request.
I thank the organizers and thanks to all of you for the patient.
And now I have a proud privilege to welcome Professor Anthrasin, sir. I hope everybody knows Professor Anthrasin. He is a big name in plant biotechnology, and he is the leader in plant genomics and genome sequencing. When in 2000, India participated in Rice Genome Sequencing project as a partner of World Consortium. Dr. Anthrasin led the Indian effort along with Professor Atlesh Tyagi from University of Delhi South Campus. So we are privileged, and you guys are basically fortunate that you are getting opportunity to listen to Dr. Anthrasin, sir, in your early research career. So with this, I welcome sir for his talk.
students हैं?
सर मोस्टली सभी स्टूडेंट्स हैं मोस्टली PhD स्टूडेंट्स ये थे तो हैं जो MSc स्टूडेंट्स
ये वाला है ये स्ट्रीमिंग कर रहे हो यूट्यूब में? यूट्यूब पे भी जूम पे PDF से नहीं चल पाएगा? चलता है अगर उसमें वो उसमें ऑडियो काम नहीं करता इससे वो करना पड़ेगा स्लाइड मूव करना हो जाएगा पहले से talk about many genome projects which have come, which have been completed in our institute as well as in other institutes in India. And then what are the challenges and way forward. And in between I will talk about what is the relevance of this genome information for genome editing. It's very, very crucial to have the genome information. So how many of you know the definition of genome? Can you raise your hand? Genome, what is genome? Okay. So it is very simple actually, because all of us have genome. And every living being, they have the genome, that is their genetic code, starting from the virus to human being, most simplest to most complex. And this definition was given more than 100 years back in 1920 by a German botanist called Hans Winkler. Because at that time we did not know about DNA, but we know that there is a genetic material. So he defined that the complete set of genetic material present in a single haploid cell of an organism. That is, this one complete set of genetic information present in the in a single cell, that will include its cytoplasm as well as the organelles like chloroplast and mitochondria. So thus one full set of information is called the genome. That includes gene as well as the non-coding regions, also repeat sequences and complete set, one complete set. Sometimes people say sum total of the genes, that is the incorrect definition of the genome. So complete set of genetic information present in a single haploid cell of an organism is the genome. And what is genomics? Genomics is the study of the genome. Study of the structure and function of the genome is called genomics. There are so many branches of genomics are there, so I will not go into that. We will just come straightway through the what is the use of genomics. Why we study the genome, why we sequence the genome, it is the structure and its function. Because it's the complete genetic code we understand, then we can utilize it. So very first point is here is our very basic curiosity, the basic knowledge which all of us want to know, our place in the universe, whom we are, where we have come from, where we are going, and what is our relationship with the other living beings on the earth. So through the process of evolution, how we have evolved. So we all know the theory of evolution. There are many kinds of theories. But once we have the complete physical map is nothing but the DNA sequence. And the genetic map, what is genetic map? Genetic map is based on the recombination frequency. That is an abstract information. Genetic map is not real, it is an abstract thing. Because you cannot touch genetic map. You can touch the figure of genetic map, but not the actual genetic map. It's a information based on the recombination frequency. But the physical map, the DNA, actually you can touch. You can take in your hand the DNA and you can see with your microscope, pure DNA. So that information is coded in there in ACGT. So once you have the complete genetic map, that means complete genetic sequence of different species, different organisms, then you can start to understand the relationship, what is called the evolutionary and phylogenetic relationship. How the different species on earth, they have related to each other, how far back they had common ancestors. All the informations are very beautifully revealed in a great detail, that how far back, like human and primates had the common ancestor, how far back rice and wheat had common ancestor, how far back animals and plant had common ancestor. So it all goes back to several billion years ago, when the life started as a single cell, and then from there it has evolved slowly and diverged into so many branches. All these relationships can be really constructed in the form of a tree of life based on the sequence data. So that's very, very useful information for basic curiosity of our origin and place in the universe. Apart from the basic curiosity, there are practical usefulness of genome information. The very first one is we can create high density DNA fingerprints of our biodiversity, our germplasm resources. Why do you need to do it? Because for the IPR protection. You know about the DNA fingerprinting? It is used in the patent disputes and criminal cases, in case of humans, in the court cases also, it has been used just for the positive identification of any individual based on the DNA fingerprint. Similarly, any life form can be identified. It can be used in the diagnostics, it can be used in the IPR disputes also. So nowadays our germplasm resources, the biodiversity is a wealth. It has been recognized by international treaty that the biodiversity, the germplasm resources actually belong to the people who have conserved them, who have protected them. But then if somebody takes it and utilizes it, they have to pay for it. Then only it is wealth, otherwise it can't be called wealth. If you can exchange it for money, then only it is wealth. But then how will you know, how will you claim that this material belongs to you? How will you prove it? So very similar to the patented dispute, people use DNA fingerprinting to prove that this child belongs to me. Similarly, you can prove that this particular variety of any plant or wild species, it belongs to us. So we need to have a very high density fingerprint. Every chromosome, regular intervals, you need to have it, because if you have a very low density fingerprint, then people can utilize it and breed your marker information out and take away 99%, remove that 1% which you are using as a marker. So once we have the genome information, we can develop such a high density fingerprint that even if they take a part of the chromosomes, we can go to the court and tell that this part of the chromosome they have been stolen from me and this belongs to me, so they have to pay for it. So that is the value of the IPR protection. It is basically to claim the property, claim your right. So genome information is useful there. The third point is that in the process of gene discovery, these are QTL mapping is the to identify the part of the genome where a useful gene belongs. And then gene discovery means actually finding out the which gene, because genome are very vast and it has thousands of genes. So once we have the genome map, the genome sequence, then that helps us to find the gene which are responsible for a particular trait. It is very similar to having the GPS in your mobile phone. Once you have the GPS, then you can very easily find the places people to go. So having the genome information is very similar to that. Once you have the genome information, you can very easily find the genes responsible for a trait. That process is called gene discovery. Out of thousands of genes and non-coding sequences, how you identify that this is the particular part of the chromosome or particular gene which is responsible for the trait. Without genome information, it will be very difficult, like looking for a needle in the haystack. Now the fourth usefulness is that, so I am coming from very basic to very practical. So the fourth one is actually the breeding process. The plant breeders, they select particular genotype out of so many thousands of material. They will select the best material. That is called process of breeding. Out of selecting so many individuals in a population, selecting the best individuals which will perform and then qualify as a variety. So this process is called selection, which breeders do. So this selection is can be aided by marker assisted selection. If you have the markers, DNA markers linked to traits, then instead of the growing the plant to the, suppose you want to do breeding in mango, instead of waiting for 5 years to see which plant is better, you can select at the seedling stage itself and then take forward only those plants which have all the required genes. So that is, I am giving one example, but it applies to any species, any plants, and I will show you example how it accelerates the process of breeding. You can do it without that also, because the breeders have been doing breeding without DNA markers also, but that is a slow process. Just like you want to travel from here to Ghaziabad, you can either walk, go bicycle, or go car, or you can go by helicopter. So the purpose is same. The breeding process is that develop a new variety, but the tools have become more refined and more efficient. So breeding without marker is also possible, but it will take long time and it will be slow. And using DNA markers, it is fast and more precise. That's the only difference. And then the last one is what this course is about, the genome editing. This is genome editing is nothing but the latest method of breeding. Like earlier people used to select, then crossing, and then create the population, then select from there, then marker assisted selection is there. In genome editing, we can simply change the gene which we want. Either you can add a gene or change the existing gene very precisely, only that gene which is responsible for the trait. But before you can go here, you need to know the gene and you need to know the genome. Both information are must. These are prerequisite for genome editing. We should know about the genome and we should know about the gene which we want to edit. Without that, your editing will be not successful, it will be imprecise. So I will come to that one by one. So again here I am going to give you a brief overview, bird's eye view, So don't worry about the too much details, because other one may have good quality, other one may have, flooding tolerant, other may have salt tolerant, disease resistance. So these genes are present in the germplasm resources. What is germplasm? Is the traditional varieties cultivated by the farmers, or old varieties, or the wild relatives of the crop plants which can be easily crossed. So good genes are present in all those things. So how will you identify? You have to screen for that. Suppose if you want to find a gene for a particular disease resistance, then you take thousands of varieties and artificially inoculate with the disease, and then you find one variety which has disease resistance. But we may not have other things which are good. But then that is called trait discovery. We find the source of the donor varieties which have a particular trait. So this is the first step, because the genes are present in the germplasm. Nobody has ever created a new gene. And of course there is no need to create also, because all the genes are present in the nature. And whatever we synthesize artificially, we simply copy what is present in the nature. Genes are present, they have evolved over millions of years, because the plants and animals, they have adapted to different situations, and their genes have evolved there. So we have to just discover them. We don't have to create the genes. It is just why it's called gene discovery, not gene invention. So the second part is gene discovery. So the gene discovery is just like looking at the one needle in a haystack, and that's where the genome information is helpful. And for that we need to create the population, structured population. That means where that particular trait is segregating and its parent is known, like a biparental population. There are so many different types of population. I have given abbreviation here. You can take these slides and read it. But because of time shortage, there could be association mapping panel, it could be F2 population, it could be real population, double haploid population. It itself make one lecture what are different types of mapping population. But in nutshell, if you have a structured population, that means you know the parentage, and the trait is segregating in a certain way, and the DNA markers are also segregating. So we look for the co-segregation. That means wherever that particular marker goes, the trait also goes. So that is called association, and there are many different software. So that's why we call the genetic mapping. We map it, then we fine map it, and then slowly we go close to the gene, and now we have the cloned the gene, and we prove it. How do we prove it? By genetic transformation. You had the lecture earlier on. So once you discover the gene, then you have to prove it through transformation. When you add that gene, the trait is restored. So this is all the very lengthy process, but this is the second process: trait discovery, then gene discovery, then they call allele mining. Allele, what is allele? You know what is allele? Yeah, these are the alternative forms of the gene. A gene is responsible for a particular trait, but one gene has several forms. That means even a single base is changed in that gene, that will make a new allele of that gene. So in the Mendelian genetics, Mendel talked about two alleles, dominant and recessive alleles, because at that time these things were not known. He had some unknown factors. He has described only two alleles, dominant allele and recessive allele. But now we know, after so much 100 years of progress, that the genes are actually sequence of DNA, and each gene can have hundreds of alleles, because even a single base pair change, addition, deletion, substitution, whatever the change may be, it will create a new allele, and the two alleles could have a different effect on the trait. So this process of finding the best allele is called allele mining. So how we do allele mining? Now we know the gene, we can amplify using PCR. I know all of you know PCR. So instead of, now you don't need to clone the gene, you can amplify. So we go back to the germplasm, the set of germplasm where the gene is varying. Then you amplify that gene only, you don't need to do the whole genome. And then you sequence it, and then see the correlation between the variation in the gene sequence.
Recording in progress.
So I was talking about the allele mining. Once we have discovered the gene, then we can look for the what is the best allele of that gene, because each gene has several alleles. So we can amplify, sequence them, look for the sequence variation and how the variation in the sequence is correlated with the variation in the trait. That helps us find the superior allele of that. So you can see then we are really ready for the molecular breeding. Not only molecular breeding, then only we are ready for the genome editing also. Genome editing is also a form of molecular breeding. Either marker assisted breeding, backcast breeding, genomic selection, and genetic transformation, or genome editing. All of that requires the gene information. Which gene we are going to change or introduce to get the desired results. And that is process called molecular breeding. Molecular breeding means it is a selection process out of a big population. What is breeding population? So each of these populations are different. Germplasm is a random collection of material, then structured population, then core sets, then breeding populations. And these tools are applied. Of course, for the genome editing, you need only one recipient and the donor gene. So it is very simple in concept but difficult in practice because more technically more advanced things are needed. But end result is same. You will end up with a variety which have all the good traits. It will have a good yield, high yield. It will have resistance against all the diseases and pests, and it will have high superior quality. So this will be accepted by the people, which is called the stakeholders. Who are the stakeholders? If I make a good variety, then who is the stakeholder of that? The stakeholder will be farmers. Yeah, that is the mistake we make, that the farmers is stakeholder and we are all working for the farmers, but that is not strictly true, because farmer is not the only stakeholder. We are also a stakeholder, and also the industry, also the consumer, whoever eats that material. So this is called value chain. From the economics people will know that from production to consumption. The person who produces the material, who processes, who transports, which market sells, who stores, and then who cooks in the kitchen. If you develop a variety of pigeon pea which takes 1 hour to cook, then the person who cooks, that person is a stakeholder. He don't want that variety, he wants a variety which cooks very quickly. Okay, and then when person who eats, if the taste is not good, they will not like that variety, so that person is also a stakeholder. Similarly, so from producer to consumer, everybody is a stakeholder and everybody has some preferences. Everybody wants a particular trait. If you want to have a mango, it should look attractive to you, it should be having long shelf life, it should be tasting. So different people have different requirements. Okay? So then breeders have to take care of all those requirements. That means you have to go to the society, conduct this survey, that what is required, and that should become your objective for gene discovery and the breeding. What happens quite often, we don't care about that thing. Just like all of you said that farmers we are working for farmers. That is not true. We are working for ourselves also, for the whole society. And all these stakeholders' preferences are important. We should decide the priority for our research priorities. Not only someone like me that tell, oh, leave, this one is not important, that is important, you work on that. I am nobody to tell that. So there should be structured survey, feedback from the stakeholders. Stakeholder, I am part of the stakeholder, but I am not the complete whole stakeholder. So this cycle comes back here again. You will discover the trait, and again it's a continuous process, keeps on going. Some traits are long lasting, like if you breed for drought resistant, that doesn't break down, but mostly disease resistance and things, they will break down because there is a co-evolution of the pathogens also. So then again you will not lose your business, because the problem will be always there and you have to keep on providing solution. So this is called the co-evolution of the host and pathogen. Even if you breed the best resistant variety today it will break down tomorrow, and then again you have to find a new solution for that. Okay, so that was a bit of an introduction, and I spend more time on that because the basic has to be very clear. The objective and the aims are the most important things. Whatever you do, your aim is the most important thing, and that is you have to keep in your mind all the time for whatever work you do, because if you lose sight of the aim, then you lose the way also. The rest of the things are the details. So now let's start the plant genome sequencing journey. In India, it is started with the RISE Genome Project in 2000, year 2000. In this institute, played the leading role along with the Delhi University South Campus. So you know what is the genome, what is its usefulness. The genome revolution was taking place around the world in 1990s, when Human Genome Project and Arabidopsis Genome Projects, people were working on, the private companies as well as institutions, through international collaboration. They were working on, but that time India was not a partner. So that after 2000, when the Arabidopsis and human genomes were published, then only we were actually awakened. So actually we were late in joining. So we were not prompt and to join these international revolutions which are taking place. So nonetheless, it is always late, but it is never too late. So we joined in 2000, and then we have caught up now with the world. So we started with the RISE genome. We become partner of the international consortium of 10 countries, and we took the challenge of sequencing chromosome 11. So that means here each partner is qualitatively they are equal, but quantitatively only they differ. Like Japan sequenced 5 chromosomes and UK sequenced only one part of a chromosome. Some sequenced 1 chromosome, we sequenced half a chromosome, but we all have to meet the common standard. That means this sequencing was done by clone, which I will describe to you little bit later. So we have to meet all the standards of international, because what we are sequencing other people are not doing. It is a complementary kind of exercise to complete the RISE genome. Why we need 10 countries? Because it was very costly at that time. So this RISE genome project was 50 crore project, 25 crore here, 25 crore in South Campus. And we sequenced only 1/2 of a chromosome. So that time it was costing roughly $1 per base pair. So RISE genome was 400 million base pair. So the project cost roughly $400 million in 5 years to complete the RISE genome. Now it is 1% of that cost, less than 1% of that cost. So this is the approach here. How we do the genome sequencing. All of you know, presume that how the sequencing is done. The Sanger's method of sequencing, Maxim Gilbert method of sequencing. Mostly the Sanger's method of sequencing was used for a very long time, but now newer technologies are used. So I will tell you how that technology has evolved. So that time we were using the Sanger's method of sequencing, using the capillary electrophoresis. That was automated sequencing, but we have to clone each fragment has to be cloned. But sequencing, you need a template DNA, you need a primer, and you need the DNTPs and a polymerase, and you should be able to read the sequencing ladder. So as an approach, see, the genomes are very big in size. There are several million or billion base pairs, and the sequencing we do, we can read only 500 base at a time with the available technologies. Even today we cannot read very large reads. Something like Illumina, you can read only 100 base pair, but with the PacBio, I will be coming to that. You can have a longer read, but it's still much smaller as compared to the whole genome. That means you have to first break the genome into small pieces, sequence them, then put it back together. It is very similar to joining a jigsaw puzzle. You have taken in jigsaw puzzle, there is a big picture, then you break into pieces. It is broken into pieces by the company, and then you join it based on the overlap. You find out what fits where, and then you join, the picture is complete. Similarly, here this is the genome. It is all ACGT, so there is no difference. You break into pieces, then you sequence 500 bases at a time. Then based on the overlap in the sequence, you can join them together. If this overlapping sequence, that means it is a common, so we can join them together and make to one piece. Similarly, you can keep on extending that and go to the entire chromosome. So this is like a brick by brick house is built. Similarly, you can just take the clone by clone sequencing. And this is a very tedious job because just compare with the jigsaw puzzle itself. If the jigsaw puzzle picture is small and pieces are big, then it will be very easy to join. But if the picture is very big and pieces are very tiny, then it will be very hard to join. Why it will be hard? Because the same tiny piece might fit at more than one places. So you will have hard time searching for that. So for the small genome, we go directly as a whole genome sequencing. But for large genomes, we go in a two-step process. First we make a large clone, and then from each of those large clones we make sub-library. So the large clones are actually back clones, bacterial artificial chromosomes, where you can clone 100 kb DNA. In the plasmid, you can clone 2 to 5 kb DNA. So in case of rice, because rice was relatively large genome of 400 million, so we followed this approach. Back clone first, the back clones were prepared, and then each back clone was sequenced. Then how do you know that which back belongs to, which chromosome and which back to sequence? So without sequencing also, based on the just fingerprinting, you can create a chromosome-wise distribution of the back clone. That is called fingerprinted context. This is how it is made. You prepare the back library of thousands of clones, then each back is digested with the restriction enzyme. Suppose EcoR1 or take any restriction enzyme, then you will get a pattern, fingerprint, which is a unique because restriction enzyme cuts at a very specific position. So each back will have a fingerprint. So just like the sequence overlap helps you to make a contig, sequence contig, so fingerprints also there will be common fragments. If there is something common between two backs, it will have a common DNA fragments. So based on the fingerprint overlap, we can create fingerprinted contigs without sequencing. And that's what this thing is here. And those fingerprinted contigs, they can be anchored on a chromosome based on the genetic map. So all these markers here, they are actually nothing but DNA sequence. And if you hybridize with the back clone, then you know that which marker hybridized with which back clone. So it will be clear in this picture here. So this is the combination of the back fingerprinting as well as genetic anchoring. So the thousands of back clones were fingerprinted, they were digested with restriction enzyme, and based on the overlapping fingerprint, they were arranged like here. So there are more than 300 clones, but they are all overlapping. That's why they put one underneath each. So there is a lot of redundancy. So you can select the minimum number of overlapping backs. Out of these 300 backs, you can select maybe hardly 10 to 15 backs, which will complete this region of the chromosome. So each back is about 100 kb. So if you take 10 back, it will create 1000 kb, that means 1 MB. So similarly, you can create all the chromosomes. And which chromosome this belongs, you take this marker sequence and hybridize on the filter. The backs are spotted on the filter. In one filter, 18,000 backs were spotted using robotics. And then you hybridize, then tell that this particular back belongs here, this belongs here, and the rest ones are arranged based on the fingerprint overlap. So this way we create the genetically anchored fingerprinted contigs. So now we can select the backs belonging to a particular chromosomes, and this is how it was done. Then the backs were distributed. For each country, a set of backs were distributed, and then we sequence. Then we go back to that making the library and then joining the jigsaw puzzle. So each, you see this is a single read here, 500 base pair, and that's how thousands and thousands of clones had to be sequenced. Each country sequenced their part, and then the rice genome was assembled. In 2 years, we created the draft of the rice genome, and it was launched in Japan in 2002 in Tokyo. That was a major event. We got letter from Prime Minister also, Prime Minister Atal Bihari Vajpayee, and this sequence was it took another 3 years to finish it. The draft was in 2002 and the finished sequence was in 2005, because just like you writing a book, first you write a draft and then you do lot of correction, make it all the gaps and things. So I'm not going into detail, but then it was published in 2005. This was the first crop genomes which has been published. Before that, Arabidopsis was published, and then this is the rice was the first one, and it has a lot of utilization. It has been used by several people now. Several varieties have been developed, and I will show you some examples here. So this particular paper has been quoted more than 5,000 times, cited, but actually it is used almost on the daily basis. Many times it is used but not cited, but at least more than 5,000 times it has already been cited also. So now you know how the rice genome was done, and from India, our two institutes, our institute and the Delhi University South Campus, we partnered to complete the job. In fact, we completed it ahead of the schedule because the funding etc. was very comfortable. It was very nicely supported by the government, although we have to create right from the scratch all the laboratories and other things. Then we received the training like Dr. Mahapatra, Dr. Tia Sharma and Dr. Kishore Gaikwad. They were sent for the training in international laboratory for 6 month training, not 10 days training. See, you have to medium term, at least 3 to 6 month training, you can learn something hands on. So they learned it and then they came back, and then I was establishing the laboratory and things. I was only attending the meetings and conferences. So we all together, there was a large group of team, several RAS, RF technicians. So we completed the job. Then once we were now expert in genome sequencing, so we were given this job of tomato genome sequencing. Once again 10 countries has joined hand because there was no change in technology, same old technology, $1 per base pair. So one country doesn't want to put all the money, so the 10 country people together can put the money and then complete the genome. This genome becomes a public property because everybody can use it for utilization. So this time we got the full chromosome 5 of tomato, and the tomato genome is 2.5 times bigger than rice genome. It is nearly 1000 MB, 950 MB to be precise. So because of the cost consideration, it was decided to sequence only 20% of the genome, only 220 MB gene-rich region. So the chromosomes have euchromatin region and heterochromatin region. So heterochromatin is highly repetitive, they are gene poor, and euchromatin is gene rich. So here in the middle of the chromosomes, if I can go back to this picture, this gray area here, 80% of the genome is in here, and that is repetitive, it is heterochromatin, gene poor. The orange one, 20% is the gene rich, is called euchromatin. So it was decided to sequence only this euchromatic region, and the bags were identified in the same way and then distributed. But there was a problem here because of the repetitive nature of the genome. Sometimes we were given wrong bags, because we were given a bag thinking that it belongs to chromosome 5, but actually it belonged to some other chromosome. So there one more step was introduced. You confirm that bag belongs to you before sequencing, because sequencing was the most costly part. The one bag will cost maybe about more than 10 lakhs. So you do the bag fish, you take a bag which is known to be on chromosome 5, this red one here, then you take the unknown one, which was supposed to be on chromosome 5, and doing back piece, then you know they both belong to the same chromosome. So now we take this back and sequenced. So it was a very slow process. And how do you know it is a slow? And you don't know it is a slow process until you have a faster method. Like you don't know the bicycle is a slow means of communication or transport unless the car was invented. So in 2008 we realized that it was a very slow, we were doing a very slow method. Okay, it was a fast method for that time, but in retrospect, in the hindsight, it became a slow method. So it was a long clone by clone approach, was a long winding path. So this particular work was stopped here, and one paper was published as a draft sequence, incomplete, because these new methods have come, what is called next generation sequencing, what we call NGS nowadays we call. So in about it started in 2005, 2006, but in about 2008 it became widely available. So there were 3 different technologies. So as compared to the older method of Sanger sequencing, this was like a super highway. Like if you compare, that was a bicycle, this was a motor car. Okay, if not the jet. So there were 3 technology. One was called 454 or pyro sequencing. So there is no time to go into too much detail because you don't have that much of time. So this 454 is called pyro sequencing. Then the second was the Illumina or Solexa. And the third one was solid, ABI solid. So their principles were different, but one thing was common in all the 3, that now we don't need to clone the DNA for sequencing. So the cloning was replaced with PCR amplification. So cloning, we are not doing cloning in bacteria now. We are cloning it using PCR on the spot, like on a glass slide, on an inert bead. The DNA fragments were immobilized and they are amplified using PCR. So each bead has one particular DNA fragment which has been amplified. Similarly, in this case, on a glass slide, each spot has a DNA fragment which is amplified there itself. And similarly here also, uses the bead here. So the chemistries were different, but the common thing was that the cloning was not needed, so that it became very faster. Now you can sequence thousands of DNA fragments in parallel. So it is called massive parallel sequencing. The output increased. So in this case here, you add one nucleotide at a time, and this is not a dideoxy one. So if there is a, suppose you are adding an adenine, so if the complementary base is thymine, then one base will be added. But if there are 2 thymine TT, then the 2 bases will be added. But the next base is not added because you are adding one nucleotide at a time. So if a single base is added, you get a light signal this much. If there are 2 bases, you get twice as much, like that. So at each position, then you take the image here based on the imaging, that how much light was emitted after adding A. From one position, this much light was emitted, that means 1A was added. If more light emitted, that means 2A were added, and likewise. And then you wash it off, then you add the next nucleotide. So this way you can construct the whole genome. And this light was emitted because of a sequencing reaction, pyrophosphate. You know that whenever you add a nucleotide, a pyrophosphate is released. ATP, out of triphosphate, one phosphate is included in the growing chain and biphosphate is released, and that can be used in the luciferase reaction, what is present in the firefly, and the light was released. That's why it is called pyrosequencing also, because of the pyrophosphate. Then in case of Illumina here, they invented a technology called reversible terminators. So here all 4 bases were mixed together, but they were labeled with 4 different color dyes, similar to the Sanger sequencing. So you use all the 4 bases, but it's a single nucleotide extension because it's a terminator technology. So you can know which color got hybridized which place, ACGT, 4 different colors, and then a single base will be added, and then it will be reversed. Then you do the treatment, then the termination is stopped, then you can add the next base, again single base. So simply by seeing the color, that which base was added, which color after which color, and then you get the sequence. So this was the Illumina sequencing. Similarly, the solid base sequencing, it was a very slow process. It is a dye-based chemistry. When the two bases are there and they ligate, they will emit a particular color light. So this was ligation-based chemistry. If a complementary base is there, it will ligate, and then this quenching and fluorescence are there. When the ligation takes place, then the fluorescence will be released. So using three different methods of detection of the addition of bases, these three technologies were developed, and they are called the NGS technologies. All three were, initially they were all popular, but after some time, only the 454 and the Illumina, they remained for a long time. Now 454 was having advantage, it has a longer read length. Illumina has a short read length of about 100 base pair, which is still a 100 base pair, but its throughput was more. And in the 454, it started with the 300 base pair, but finally it became 2000 base pair. And 454 technology, it replaced the Sanger sequencing, because Sanger was still popular because of the longer read length. So longer read length is very important for what? For assembly of the genome. I gave you the analogy that if the jigsaw piece is bigger, it will be easier to join. So the longer read length means longer piece of jigsaw. So I will come to that again in the third generation sequencing, how that is revolutionized the sequencing. But at that time, in about 2008, you can see this is the Sanger read length here, much longer, then followed by 454, then the Illumina, and then solid was even 50 base pair only. But the throughput was highest here. Shorter the read length, higher the throughput. But then more difficult it is to assemble. So we had to, choose and mix different technology to sequence the genomes. The tomato genome, which we started trying to sequence only 20%, after this NGS came, then it was sequenced to 100%, nearly 90% here. And each chromosome was sequenced to completion. Both this is the heterochromatin in the middle, you can see the repeat sequences, and the end, the genes were at the end. So this is one chromosome. Similarly, all the chromosomes were sequenced and published in 2004. This is also a very good resource, and it is being used worldwide. Then the next comes the wheat. Wheat is a very big genome. It is 40 times bigger than rice genome, and it is 6 times bigger than the human genome. Human genome is only 3 billion base pair. This is 18 billion base pair. So that's why international community, and also it is a rich man's crop actually. Wheat, rice is poor man's crop because 90% of the rice is grown in Asia, India, China, Southeast Asia, and consumed. That time China was poor. Now it is one of the richest country. But rice was a poor man's crop. And actually Japan was the richest country or rice consumer, but there are not many people in Japan. But because of that, Japan contributed 50% of the rice genome. So only those people will contribute for whom the crop is important. That is called stakeholder. If somebody who doesn't eat rice, why they will pay a single dollar for sequencing rice, unless they have a business, to selling seed to you. So similarly, stakeholder, the wheat has a rich stakeholder, like Europe, America, Australia. Their wheat is their number one crop. But this genome is so big that even for the rich people it was a very daunting task. 6 times human genome. So they were looking for a partner once again. They approached us many times also. But after some times we became partner. And this time we got this sequence, chromosome 2A completely. The PAUs, Ludhiana and South Campus and our institute. Again we joined hand. And you can see that This one arm is the whole rice genome. 2A short arm is complete rice genome. 2A long is one more rice genome. So 21 chromosome, roughly 40 rice genomes. So one chromosome arm is equal to one rice genome. And this we sequence using the NGS technology, of course, using the Illumina and the 454. We completed. I'm not going into detail. Only thing I will tell here that how it was possible to share the chromosomes, because now it was not necessary neither possible to share the chromosomes unless you can separate the chromosomes. Because in the whole genome sequencing, when you are using NGS, you have to take the whole DNA and sequence it, because no back cloning and chromosome by chromosome distribution of backs. In case of wheat, it was possible because wheat has a special genetic stock, special cytogenetic stocks, which other crops do not have, which is called double ditelocentric. Double ditelocentric means, telocentric means when a chromosome has centromere at the end, that is called telocentric chromosome. So for each of the 21 chromosomes, one scientist from University of Missouri in USA, he created genetic stocks where one at a time each chromosome was in two parts, short arm separate with a telomere, long arm separate with a telomere. So this is called double ditelocentric. Otherwise genetically it is identical to the normal wheat, but one chromosome is broken into two pieces. Because it has the centromere, it can be maintained. So because of the size difference, telocentric chromosomes are shorter than the full chromosome. So you can see they can be separated using the flow cytometry, laser assisted flow cytometry. The chromosomes were separated and then chromosome specific DNA was distributed. This work was done by a scientist in Czech Republic, very small country, a scientist called Jaroslav. He actually prepared for the entire world, and the 25 countries they joined hands together to sequence the wheat genome. And in 2014 the draft was published. That this draft was still very incomplete, only 60% of the genome, but it was chromosome wise sequence, because it was possible to isolate the chromosome specific DNA. So even though it was very highly incomplete, but it was arranged into chromosome wise. This is called the chromosome based draft sequence of hexaploid wheat. 25 countries including some private company joined hands to complete this job. Then it took another 4 years. I showed you in the rice genome it took 3 years from draft to reference genome. Here it took 4 years from draft to reference genome, despite the advanced technology. And then even this draft is nowhere, this reference genome, which was published in Science in 2018, it is nowhere near the rice reference genome. This still has a lot of sequence gap, about 2% N is there. So slowly, one by one, people are taking one chromosome at a time, like France is working on chromosome 3B, and they are completing. We actually left this thing, we left the 2A to international community to complete. We washed our hands off it because it was quite expensive also, and then these sequences are now available in the public domain. And the other thing is that this is a concern of all the rich country. Okay, so this is available anyway, so why we should spend a lot of money on doing wheat? Because we have so many other crops which other people don't have interest. So I will come to that how we have to prioritize to generate these resources. So this is the completes the genome sequencing of rice, tomato and wheat through international partnership. And you have also seen that how the transition took place from the Sanger sequencing to NGS sequencing, and how it speeded up the process, and also the cost became less. It became almost less than 1% of what it was at the time of the rice genome sequencing. So now we come to the next step. I will come to the third generation sequencing when I come to the mango genome sequencing. I will introduce you to how that was helpful in that. But now we come to the utilization part. Utilization is not possible without the gene discovery. Now we have the genome, but we don't have we know about 37,000 rice gene based on the in silico gene prediction and annotation. but for one particular trait, which is the gene, we don't know. That is the gene discovery process. And we focused on this high grain number, basmati quality, and the salt tolerance. 3 traits we started here. Similarly, many people started different traits. All over the world, there were hundreds of genes have been now discovered, because gene discovery is a slow process. Now in hindsight, genome sequencing is easy, but gene discovery is more difficult because it needs collaboration between many different disciplines. You need collaboration between breeder, physiologist, geneticist, and agronomist, physiologist to get to the gene, biochemist. So I will cut the long story short because I have already introduced to you how we go from the trait to the gene. So this is the trait here. Now to go to the gene, mapping, fine mapping. Mapping means find the genomic region where the gene lies. It is on the chromosome. This is one example for high grain number. I will give you only one example because every trait is discovered in the same way. Gene for any trait will be discovered in the same way. The process is same. So this long arm of chromosome 4, we found this major gene, and this is the gene of protein kinase domain containing gene. This particular gene was responsible for this high grain number. Now we have transferred this one into 12 different varieties, mega varieties of rice. You know what is mega variety? Mega variety means if a variety is so good, so popular, it is cultivated in more than 1 million hectares. So that is called mega variety. It is very popular among the farmers. But even it is a mega variety, it will have certain defects. Just like even Sachin Tendulkar will have certain demerits, or even some big people also will have certain demerits. So it is good in so many things, but still it needs improvement. We cannot improve people, but we can improve varieties, right? So this is we took 12 different varieties and we transferred these genes for high grain number, and we found that there was significant grain in each of those varieties because by transferring this good gene. So this was done by backcross breeding. All the breeders, people who are breeders, they will know that what is backcross breeding. You transfer a good gene from an inferior background into a good background by repeated backcrossing so that you can recover the background and add the useful gene. So marker assisted backcrossing means what was done, basically you are creating a isogenic line, near isogenic lines basically, so that the one gene is added in a good background. So we did that. Normally it will take 7 to 8 backcrosses, it might take 5, 6, 7 years, but through marker assisted background selection and foreground selection you can do it in 3 years. So this was done. This is the foreground marker for that high grain number, and for the background we designed a chip here in this institute. This is the patented chip. So we use 50,000 SNP markers representing the whole genome. So this is one of those backgrounds, 1121, famous basmati variety. We transfer this high grain number, and blue color means we recovered the background using the 50K chip, and the foreground using the foreground marker, maybe SSR marker or maybe any kind of marker linked to the gene. So similarly in all the 12 varieties you can see this panicule size has increased. This is the original, this is the with the QTL. This is the original, this is with the QTL. So in all the backgrounds, I will show you one in detail here. In Samba Masuri. Samba Masuri is a very popular variety. This is the original Samba Masuri. When we transfer this one gene through marker assisted breeding, the panicule size becomes more than double. The number of grains here see 200 in the original Samba Masuri, but with that gene it becomes more than 400. So ideally the yield should double actually, but yield did not double because the number of tillers was reduced and some other things, but still there was a 15% increase in yield, and this has been nominated in the ACRIP trials and it is being taken forward for release. You can do this similar thing with the genome editing also. I will come to that, how genome editing is related to backcross breeding. Genome editing is basically a new form of backcross breeding. So let us come back to again here. the concept of back-cross breeding. This is basically we are doing the developing the nils or near isogenic lines in the mega varieties, the varieties which are already very popular, but they may be lacking some disease resistance or certain traits for drought tolerance. You can add that in that background, then it will become a nil of that mega variety. So these mega varieties are very popular among the farmers because they are high yielding. In contrast, the traditional varieties which have high level of stress tolerance, they are good quality, but farmers do not grow them now because they are low yielding. So even many people say that we lost our old varieties, green revolution has done harm to us because we lost all the varieties, but that is not true because all the varieties are there in the NBPGR gene bank. More than 1 lakh rice varieties are there, but farmer will not grow it because they are not high yielding. So what you have to do, you have to combine the high yield with these traits. That's what this program was there, which I am going to describe. So basically you are developing a mega variety nils near isogenic lines through marker assisted breeding, and very quickly we have released so many varieties now. Most of the varieties which have been released through marker assisted breeding, they have used this technology, back cross breeding, like transferring one good trait into already established variety. Now here the true isogenic, but it still has some linkage drag here. The true isogenic, the genome editing can produce a true isogenic lines without any linkage, zero linkage drag. I will give you example of linkage drag here, because when you do by back-cross breeding, you are normally transferring 1 to 3 MB, sometimes 4 to 5 MB of genes, because you cannot recombine a single gene. There will be maybe hundreds of genes which are tightly linked with that. That will also go along with that. So this is one example of this Swarna sub1. Swarna is a mega variety. It is sensitive to flooding. If it is flooded in the water, submerged, it will die completely. But when the sub1 gene was transferred, the gene was cloned using the genome information. Within 3 years, 2006, gene was cloned. 2009, the variety was released. It became very popular. So this is process called developing nils. Nils are also called essentially derived varieties. That means adding one gene to a already existing varieties. So we developed a very big program called QTL2 variety to transfer these genes for submergence, drought and salinity tolerance into different varieties. These are genes here, drought, salt tal, then there are 7 genes for drought tolerance, 1 for submergence tolerance. And for each of those combinations, this was again a very big project. Total cost was 27 crore actually over 10 years. And there were 15 institutes were involved, so that it was shared in the network. So we developed the peak markers, and then same using this 50K chip for background selection, the varieties were released. I am just giving you one example here that on chromosome 1, there was a gene for DTY 1.1. This is a QTL for drought tolerance. So all the tall varieties, traditional varieties, they were tolerant to drought because of this particular major QTL for drought tolerance. But this QTL was very tightly linked with the gene for plant height. So when we transferred that dwarf, semi-dwarf allele from the DGO gene, which is the donor of the dwarf allele of the SD1 gene, they inadvertently, without knowing, we transferred the susceptibility allele of this gene, because you cannot break this linkage in the normal course of breeding. But with the genome information by fine mapping in this program, we were able to break this linkage. For this you have to go more than 5,000 lines. Then you found the line which were dwarf but still drought tolerant. This is the line of Swarna which has a dwarf, Swarna DTY1 dwarf. This is the original Swarna which was dwarf, but it has tightly linked sensitivity allele. So see, it is dried, but then when you recombine that, when you get the tolerant allele and combine with the dwarfing gene, then you get this Swarna is different from that. So this is a very typical example of negative linkage drag. when you want to transfer through normal breeding process, always every gene you transfer 2 to 3 MB. That means 1 MB means 100 genes. You are transferring 10 kb per each gene is about 10 kb. So 3 MB means 300 genes. I am transferring the semi-dwarfism, but from both sides I am transferring 100 genes this side, 100 genes that side. And if it happen to be a negative linkage drag like this one, then this is one example, but there could be anything, maybe quality related or any other things. You can have a problem. Of course, this program was very successful. We have developed so far 17 varieties. This says only 14, but all these varieties are very, very popular. Now let us come back to how I'm just trying to show you the relevance of this thing to genome editing. Through genome editing, basically we are creating the near isogenic lines. You take a good variety, you will be doing genome editing in good variety only, because you will be changing one gene and editing it to make it better than the earlier one, but the rest of the background has to be good, otherwise you have to do again back crossing to transfer in a good background. So better to do genome editing in a good background, so that only one gene you are going to change, tweak. What about the remaining 37,000 genes? They have to be good only, because if you do genome editing in a bad background, so nature has already done it. All the good genes are in the bad background, so we are doing back crossing, and as a result we are having a negative linkage drag. So while choosing the variety for genome editing, better to choose a good background only. This is the precaution. So what is the advantage of near isogenic lines? And if you use genome editing, you will be creating isogenic lines, perfect isogenic lines, because it will differ by only one gene, no linkage drag. So what is the advantage? They can be released as a essentially derived variety with minimum amount of testing. Variety is already popular for 40 years among the farmers, तो आपको और 3 साल testing नहीं करनी है. Just only to prove that this added trait is working. 2 years and the trials are constituted. Straightway to go to ABT, Advanced Varietal Trial. ABT1, ABT2, thus how those 17 varieties were released. Then what you can do, you can pyramid multiple QTLs and genes. If you have different nils, you can intercross them and they still remain nils. If you cross 2 nils, and then you can pyramid as many genes as possible, there is no need to do back crossing, because nil crossed with nil will remain nil. And if it is IL, if it is a real isogenic lines, it's even better, because if it is near isogenic lines, every gene you pyramid 3 MB will be adding linkage drag. Okay. So once again the genome editing will be advantageous. Then you can deploy the strategic deployment of the R genes. Suppose you have 10 different resistance genes against a particular disease, so you don't want to put all of them at once, because if you put them all at once, then the super race will develop, and then you will not find the solution of that. So strategically we develop, like if you use one particular pesticide and the insect develops resistance to that, then you can use another pesticide. But if you start using, if it develops multi-drug resistant, then it will be hard to control. So similarly, the resistance genes can be deployed. You can put only those genes in the variety which are enough for that particular pest. And the multi-line concept has already been promoted by KJ Fry long time back, but now we have the tools to develop really truly isogenic lines where you can deploy this technology. Once again, one can have location specific deployment, means use only those genes which are needed. Don't unnecessarily put the genes to have the unnecessary linkage drag or unnecessary burden on the plant. So genome editing, you may have been told about this thing. Genome editing guidelines, standard operating procedures have been cleared by the government. It has been notified. So now we can do SDN1 and SDN2. Why they were notified by the government? Because they are not like transgenic. SDN1 and SDN2 is just like I showed you the breeding up nils. There is no foreign DNA, only a particular gene you are changing, you are making a better allele of that gene. By the either you are shutting it down, if it is a negative regulator of trait, suppose it is a susceptibility gene, and then you knock it down by introducing a stop codon right in the beginning, or making it ineffective, that is called knockdown, then it is a null allele of that gene basically. So you have created a null allele of a gene which is negative regulator of a particular trait. Maybe an anti-nutritional factor could be there, a susceptibility gene could be there, or a bad color, maybe people don't like particular color, you can knock down that. So you are creating a null allele for their genes. Basically you are developing isogenic lines. You are not introducing any foreign genes, so there is no need to control like GM crops. Even though you are using transformation to start with, your end product is free from the transgene. Because genome editing is performed at a different location and the CRISPR-Cas is integrated at a different place, so you can separate it out. Then the second SDN2 is that SDN2 is like allele replacement. Like you have a better allele, you can do template based gene editing, replace the allele with another one or make some minor modification. So in the definition it has been defined, if the final product is similar to the allele of the present in the primary or secondary gene pool. That means what we are doing by crossing, I am doing breeding by crossing the two things, and as a result sometimes I get negative linkage drag. But if I use genome editing to do the same thing, I will replace the allele without having negative linkage drag. So if I take a gene from say wild relative of rice at the same better allele at the same locus, then replace it that locus with the locus from at the same chromosome position at the same locus, then it will be considered as SDN2. If you put it in another position, then it will not qualify as SDN2, even though it is coming from another rice variety. But if it is going to another location, then it will not qualify. But if it is the same locus, this has been defined in the guideline. So if you make the changes at a particular locus in the targeted locus, and if it is similar to the allele in the primary or secondary gene pool, then it qualifies as SDN2 and it does not need GM type of. In perspective, what I showed you at the marker assisted breeding, your genome editing is just simply a more high-fi version of, more efficient version of marker assisted breeding. Once again here it has been defined, the SDN2 has been defined because it has been integrated at the same position. If it is integrated at a different position, then it will be treated as a transgenic. Same gene, if it is integrated at a different position, so you have to prove that what genome editing you did, it was done at the same place. Allele replacement was at the same place. So I will rush through actually. This I want to show very quickly that in genome editing has two parts, SDN1 where mostly you are doing knocking down by non-homologous end joining, and SDN2 you are doing allele replacement basically. You are creating new allele or you can enhancing the expression. And if the genes are present in the nature, in the wild relatives or in other crop varieties, so then you can take that gene and replace that same position by homology derived gene editing at the same, that will qualify as SDN2. So we have to look for these superior alleles, and then the wild rice, which is easily crossable with the cultivated rice, is a good source of genes for different kinds of stresses because it is surviving the stress in the nature, it has widely adapted. So I collected from different places. These are some lowland areas in different part of the country, in upland areas also. This grows wildly in this particular rainy season. It is also adapted to the highly saline conditions, sodic soil, coastal salinity, deep water, as well as dry conditions. But it is highly threatened due to developmental pressure. Like some housing will come, some school building will come, some industrial park will come, or some road is getting widening. So this is under great threat. So this is a conservation is very important. So I have collected 1,000 of them from different parts of the country and studied their population structure. I am not going to detail, but I screen them for different traits. Once again, this is trait discovery, and then I find certain lines which are more tolerant to salt than the best of the land races. Like this is the Pokhali. It is tolerant, but certain wild rice are even better than that. So that means they have better allele of those genes. So I created a population in backcross one, then using this chip here, we create the genetic map. This is the phenotyping. You can see all the traits are segregating, but I am more interested in the salt tolerance only, because by backcrossing I will eliminate all those things. So here the red ones are the genes identified for salt tolerance. This is also the chromosome 1 here. This is a well-known allele of saltal1, but this has a superior allele even in other chromosomes. And then underlying genes are already, candidate genes have been identified, but they are not confirmed because you have to do transformation to confirm it. Chromosome 1, chromosome 3. Simply we are doing the backcrossing. So this is the conventional. See, now after the genome editing has come, marker assisted breeding will become conventional. As much जब marker assisted breeding आया तो conventional breeding become conventional. Now when the NGS came, then Sanger became conventional. So in front of genome editing, my prediction is that if it goes through like this, without much hindrance in the regulation, all those things, which should not, then this genome editing has the potential to replace the conventional breeding. Basically, what you are doing, you are crossing and transferring and selecting. Once you know where the gene are, you directly transfer the gene, 1 gene, 2 gene, 3 gene, 10 gene you transfer in a good background. So it will shortcut the breeding process. So breeders will slowly turn into molecular breeder, then basically you are still a breeder, but you are using more modern tools of improvement. So until and unless this breeding genome editing becomes streamlined, we can keep on using these older tools. Now very quickly, I had told you that I will take about 1.5 hours. So within 10 minutes I will finish, about 12 o'clock, okay? So don't worry too much that I will take very long lectures. So I hope you are enjoying it, because I am not going into too much detail. I am only giving you the overview that this is a very interesting technology, and how the genome information is crucial to this technology, and what is the advantage of this technology over the conventional. So it is actually the frontier technology about which we are talking. So I told you that why we lost interest in the wheat genome sequencing later on after contributing initially, because we have so many crops and species, fruit and vegetable crops, medicinal aromatic plants, which are important for us and for nobody else. So we will put our energy in doing that. So based on that logic, we selected 3 crops: pigeon pea, jute and mango. Pigeon pea, jute and mango, India is number 1 in the world, and more than 90% is produced, consumed, everything by India. And we have started, we have to import from Myanmar and from other countries. So we choose pigeon pea genome sequencing. And as expected, it was very hard to get partners. We were lucky to find one partner from Doug Cook from the University of California, Davis, but he was not funded for that. Because of his own interest, he actually tried to help us under the Indo-US Knowledge Initiative program. There was a big program called Indo-US Knowledge Initiative. That was on the sidelines of Indo-US nuclear deal. So once again, there was a biotechnology was a component of that. And then when we insisted that we want partnership in Pigeon P, there was a great reluctance from the other side. Naturally, you can understand that. Why they should put money in your Pigeon P, in your jute? It's not interesting to them, but we still were interested. It's just like the, mismatch in a marriage, it will end up into divorce. So similarly, this ended into divorce. The Indo-US Knowledge Initiative, trying to find partnership in Pigeon P, it was not interested partner were forced to get married. But anyway, because for us it was important, we carried on. And then in parallel to those efforts, we published this Pigeon P genome sequence in 2011. This is the first genome sequenced from India, totally done in India by the Indians. Any genome, whether animal or plant or microbes, this was the first one from India. Then using the genome information, we were able to map the genes for dwarfing trait here, because Pigeon P also needs green revolution, just like rice and wheat. The present Pigeon P varieties are taller than me, so you cannot use mechanical operations, you cannot spray them. Harvest index is very low, maybe 10-12%. So we need modern varieties which are this height. They can be, you can do it for spraying, etc., and you can do mechanical harvesting. So we have identified the genes for dwarfing traits. We developed the chip for Pigeon P also, nobody else will develop it. For rice there are 4 or 5 different chips are there, but for Pigeon P are not that many. Once again we have done the mapping of important traits, yield related traits and other traits. And we have developed in collaboration with IRI, some 4 varieties have been now developed which are the semi-dwarf height. And now there is a trend, now the trend has started that the future Pigeon P variety will be early maturity, they will be semi-dwarf height, they will have higher harvest index, so that you can get more productivity and you don't have to import from Myanmar or Africa. Jute is similar. Jute is mostly from only 2 countries are actually involved, India and Bangladesh. So we sequenced this variety of jute called Naveen, JRO524. This is the variety developed 30 years back, but still it is the popular variety, not much improvement because we don't have a lot of genetic information. But with the genome information now, hopefully there will be more progress in that. We have mapped the gene for fiber quality. There is some, this thing is coming, I think, I don't know how it comes. I think some people do it, mischief in the online. So we have mapped, we have a reference quality of jute now. We published the draft one, but this reference quality we have got yet to publish within just 46 countries. Now let's come to the mango, because mango was very difficult, and this will highlight you the problem for which we need the 3rd generation sequencing, and which was helpful. So mango genome size is only very similar to rice, about 400 MB, but it has high level of heterozygosity. So the genomes which were sequenced till now, they were either highly inbred line or double haploid lines. That means they were pure lines. Okay. But we know that all the diploid organisms, they are not pure lines, they are certain degree of heterozygosity, like chromosome 1 coming from the male parent and the female parent, they will be different. So in case of mango, they were very, very different, because there is a lot of heterozygosity, there is no inbred line, double haploidy doesn't work in this. Inbreeding, you have to 100 years to make selfing, and there will be so much inbreeding depression that you cannot maintain it. That information has come out from that. So as a result, what happened that you cannot assemble the genome. When you do this NGS sequencing using Illumina, the the male and female chromosomes contigs, they don't collapse into one. So when we assembled, the assembly size was bigger than the genome size. In so much so that some of our international partners thought that mango is actually a polyploid and actually its genome size is 800 MB, not 400 MB. So in some of the PAG meeting, actually they presented this thing that it is a polyploid because their assembly was going more than 700 MB. And then we actually found a solution in PAG Bio. We adopted this technology much earlier. It has been now 5-6 years back. In fact, we published the first draft of Mango in 2016. So we found a solution here using the PAG Bio sequencing. PAG Bio can generate very long read lengths. So that time 4-5 kilobase read length. So what we did a simple change in the assembly strategy, that you don't go for 100% match, because 100% match can only work when it is a homozygous line, pure line. If the male female are different, then you have to allow mismatches so that assembly can take place. So we go for 1 kb overlap and 15% mismatch, and just doing this single change, very simple strategic change in the assembly process, using the Falcon assembler, we were able to assemble. So now what we are getting nearly 492 MB, this came down to 326 MB, and as expected we were able to get this assembly. So that was the start. So what is this new technology? Is called single molecule real time sequencing. Now at present there are two technologies, one is the PAC Bio and the another one is the nanopore, Oxford nanopore technology, and this is the PAC Bio real time sequencing. So here what is the difference between the second generation and the third generation? Read length is the output, but the technology wise the difference is that you don't need to do even PCR amplification. See, the DNA template does not require cloning either in bacteria or through PCR. So you isolate the DNA and you don't amplify, straightway that is your template. Only you add the adapters both sides, and here is the circular template you prepare, but there is no amplification. You need to isolate high molecular weight DNA because the read length is quite big, both in nanopore as well as in here. So that has advantage of the assembly. That's how we allowed to get 1 kb overlap. If your read length itself is 100 base pair, then you cannot get 1 kb overlap. But if the read length is 5 kb, then we can get 1 kb overlap and mismatch. But of course now you have much better software, and that we use by logic, and then that worked. But now there are many software are there which can separate the phases. Like the male and female phases can be separated, then you can have two sets of chromosome one coming from the male, one coming from the female, and that is called the phase separated assembly. And there are software like Hi-Phi-ism, phase separated. So there are two approaches here actually. One paper came in 2018 which gave a method for trio-binning. By Sergey Koren, there was one scientist who published the first paper where you can have the two parents and the progeny. So if you have the parents are known, then you can separate the maternal and paternal reads. So the two parents you sequence using Illumina and the progeny you sequence using PAC Bio, and then you can separate the PAC Bio reads into maternal reads and the paternal reads based on the polymorphism, which is just the Illumina sequencing. And then you can assemble them separately. So initially we tried to collapse the two into one by allowing mismatches, but now we use this trio-binning approach because we were lucky actually that both male and female parents of Amrapali are there in the IRI garden. Original Neelam and Dasari, which were used to cross to produce Amrapali. So both parents are there and Amrapali is there. So using this trio-binning approach, we were able to separate the Dasari reads and Neelam reads. So now we have two different genomes, Dasari genome and Neelam genome. This is called trio-binning approach. But quite often you may not have the parental information, because many of the livestock or even other mango varieties, if you want to separate or another heterozygous species, parents may not be known. In that case, you can still separate the phases using the software approach. Using the software with long read sequences and more fold coverage, so if there are 20 pair of chromosome, you can get 20 pair one assembly and then alternate assembly, what is called the haplotype, and the alternate phase, main phase and alternate phase. So now people are coming up with this maternal and paternal assembly. Not maternal paternal, you can only call when you know the maternal and paternal parents, but if you don't know the parents, you will have the main assembly and alternate assembly. In that case, it will not be strictly maternal and paternal, but the phased assembly. So these things are coming up. And we found some interesting information there that there are a lot of genes which complement each other. Genes present in one parent but absent in the other parents, and they complement each other. So these are the routine things, like we do the phylogenetic analysis, and they say the mango is very close to the cashew. Among all the fruit sequence, mango is closest to the cashew nut. They look quite different. Then you can do association mapping. Then we have developed a chip for mango also, which is the 80K chip, 80,000 SNP. And we have utilized this chip for diversity analysis as well as for association mapping. There is a big QTL was identified for the fruit acidity. So if you can do the phenotyping, genotyping you can do either by chip or you can do by genome sequencing, GBS, and you can identify the QTLs. Then you can do fine mapping and then utilize, just like I have showed the example, how you proceed from trait discovery to gene discovery to allele mining to utilization. So the process is same in any species, but the genome information may not be available for the species of your interest. If you are working on a particular species and genome is not available, then you cannot do many of these things. Genome editing you can definitely not do it. So similarly we have sequenced the genome of Raja coaptata here at our institute. Then also the dwarf coconut genome in collaboration with the Kasargod Institute, CPCRI. Then tea genome has been sequenced by Tapan Mandal. Then guar genome by Dr. Gaikwad. He will be presenting these things. Then other institute where we are not involved, the chickpea genome has been sequenced by the Delhi University South Campus. Then another one, this is the Aja directa indica, the neem genome has been published by institute in Bangalore. Then you have the Tulsi genome by CMAP and one more institute. Then Haldi genome by ISR Bhopal. This is my second last slide, so you can relax now. So now you know that what is the importance of the genome information when we are working on any particular species. Having genome information is like having GPS in your mobile. Would you like to have a mobile without GPS? It will be very, very hard to work. So in India, just before the corona came, there was a big plan to sequence 1,000 genomes with a project of 500 crore. It was processed completely up to the final stage, but because of the corona, the sanction letter did not come because the priority changed to corona and other things. So hopefully this gets revived. This is the 500 animal and 500 plant genomes. It was actually planned. So the last slide here is that what is the way forward? What we should do? Like we are doing different things. Maybe somebody breeder, somebody plant physiologist, somebody different area plant pathology you are doing. And then anybody who is working on the living system, microbiologist, you need to have the genome information. You can forget that, you can avoid the genome information because this is something necessary information you will be avoiding. because once we have the genome, then we can do all those things. We can do basic study, we can do applied study, we can do manipulation, you can do breeding, fingerprinting, everything you can do. So what is needed to be done? First thing is the conservation and high throughput evaluation of germplasm, because the germplasm resources is very important. If you lose, like I have showed it in rice, then if the germplasm is not there, then the gene is also not there, then all the techniques will be kept in the shelf, because germplasm is the primary material on which you will be working. So we need to conserve it and then evaluate it so that it can be utilized and not kept in only the chest. Then the prioritization has to be done. You prioritize what is economically important for us, we work on that first, like we chose pigeon pea, jute, mango. There are so many, several others are there which are important, but no work is done and nobody else will put their money. So each one of you, wherever you go, there will be some institute priority will be there. This prioritization is decided there. Then we create high density map, RNA sequencing, then plant biology. This is very important. All branches of biology are very, very important for gene discovery. Without all branches, you cannot do the discover gene. Without biochemistry, without physiology, without pathology, the traditional disciplines are equally important. Without traditional breeding, you cannot use these new tools. New tools are only modern tools to supplement the traditional discipline. Then one can do gene discovery, association mapping, genomics assisted breeding, which I have showed you several examples, and finally you can do accelerated plant breeding through genome editing using SDN1 and SDN2. So that's it. If you have any question, quick question, I will be happy to answer. माइक्रोफोन दे जरा. या तो आप जोर से बोलिए या माइक्रोफोन.
This could be little of off topic, but I wanted to ask what would be the future of cereals and millet research in India?
Future of
Cereal and millet research in India.
मिलेट रिसर्च. मिलेट के पहले क्या बोला आपने? Cereals and millet. So cereals is very important. You have seen the rice, wheat, maize. They are the three most important crops to feed the world, and they will remain to be most important crops. And millets are becoming more important for the health and nutrition region. So the future is very bright. I don't see there is any. Future is related to the consumption. If you are going to eat cereal, then cereal is important. If you eat millet, then millet is important. If you don't eat it, then it is not important. We don't want to just decorate it in the in the almirah. So whatever is economically important for us, and of course strategically also for environment point of view, not only for food and shelter and fiber, and for our environment. So these are important crops. Cereal is definitely, undoubtedly very important. It has been. Because no matter how much we talk about millet, people will not give up eating, rice and wheat and maize. And the productivity gap is also there. You have to work a lot to increase the productivity and resilience. So these are the slow changes. But there may be some shift. Maybe you will be eating more millets and less cereals. But both of them will continue to be important. And then if the commodity is important, then research is also important. Because there is continuous need for improving the productivity and quality and the resilience to the climate.
Sir, for targeting specific gene for editing, will pangenomics data play crucial role?
Yes, very, very important actually. I forgot that point, thank you. Because when we sequence a genome of one particular individual, that does not represent the entire species. So in the species there are hundreds and thousands of individual, and each individual will have certain unique genes which is not present in the other individual. This is called presence absence variation. So if you want to have a complete picture, then you need to sequence more than one variety. So as we sequence more varieties, so the first reference genome is important, because the first variety has to be sequenced in very high quality reference genome, and that works as a base. On that base we can resequence more variety, and then additional genes will be added to that base, and that makes the pan genome. And so initially the gain will be more, but after some times you will not gain much by sequencing more varieties. So there's a graph goes like that. You know, maybe if you sequence 200, 300 varieties, you will get pretty much the saturation point, after which there will be no further gain. But definitely sequencing more than one variety will create a pan genome situation, where which is important for the genome editing or even for breeding, for anything. So pan genome is very important.
Sir, can we use biparental population for genome-wide association studies?
Yes. You can actually, that is again very important question, because normally we use a association mapping panel for genome-wide association studies. You will have a number of genotypes, you will assemble a panel and you do the genome-wide association study. The software for that is different, like you can use Ticel and other software. But for biparental mating, biparental population, we normally use the QTL mapping software. But now there some papers have come where they use the GWAS software for the QTL mapping, and then they get better results. Basically in both of the methods, whether you do QTL mapping or GWAS, you are looking for marker trait association, isn't it?
Yes.
You are going for the co-segregation. When a marker is segregating with the trait, then that is your QTL. So people are finding better results using the GWAS software. In that is called this single marker trait association. You can get sometimes better results. But each of the time you have to validate actually. Whether you do QTL mapping or GWAS, you identify the QTL, then you have to validate in another population or another material. Because many of the times, using the best of the software, you get sometimes false QTLs, even after doing all those corrections, or maybe it will bring very small effect. So validation, the next step becomes very important. Validation of the QTL, then fine mapping, then identifying the candidate genes. So regarding your question, you can use GWAS for biparental population.
Sir, but in biparental population, only limited variation will be present, no, sir. The variation present.
Okay, that is true, but even limited, our aim is to find the genes, ne? Your aim is to discover the gene or genomic region and then finally the genes. So if the two parents differ for a gene, even for a single gene, that is good enough exercise to find the gene. Even for the fine mapping, we use QTL nils actually. QTL nil is just differ for only one QTL. When you take two varieties, they differ for a trait, there may be more than one genes in that. But when you go for fine mapping, you are taking a QTL nils. They should not differ for any other traits in the background. So the idea is not go for more variation. Idea is to get the gene. The purpose of QTL mapping or GWAS is to identify the genomic regions or the genes responsible for the trait. So if that purpose is solved, then it doesn't matter if it is less variation or more variation. Less variation means it will be more precise, because your phenotyping is not interfered by too many things in the background. That is the disadvantage of GWAS actually, because it will have too many QTLs and each contributing very small variation. So usually, or most of the time, the QTL identified the GWAS method has to be validated by biparental population. And the QTL identified by biparental population has to be fine mapped using the crossing two QTL needles, which differ for only one QTL. So it is the process for going very broad to very narrow and finally identify the gene.
Thank you, sir.
Okay.
So if there is no further question, I would like to thank sir for nicely summarizing all the genomics work which have been done. Thank you, sir.
Hello, hello. हाँ अपन दोनों. सर ऊपर वाला.
And this is for distribution, Anil.
Yes,
You can be useful because lot of things are there. I have to skip slides.
yes.
चाय वाय नहीं चाहिए?
Sir, distribute करवा देते हैं. So our next speaker is Dr. Pranam Mandal. He is principal scientist at NIPB, and he is an excellent teacher, which you will realize now when he will start taking your class. Okay. So Pranam Mandal sir, over to you.
हाँ 5 minutes break लो.
Hello. Hello, hello. Hello, 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 hello. Hello, hello. Hello, hello, hello. Hello, hello. Hello, 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 hello. hello, 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 check, hello, 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 hello,
अकबर ये चलता है
hello, 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 hello.
ये माइक है सर, ये इस पे बाकी लोग नहीं सुन पाएंगे, उधर अगर बोलेंगे तो नहीं सुन। ये लोग सुन लेंगे सर, ऑनलाइन वाले नहीं सुन पाएंगे। यहीं पर खड़ा होना पड़ेगा।
हेलो
इसके ऊपर हवा आ रहा है, ये इसको
बंद कर।
तो गुड आफ्टरनून।
हेलो
ये वाला माइक है ना? ओके। सो सी माय क्लास इज ऑन, क्लास बोलो तुम्हारा लेक्चर बोलो, वट एवर इट इज इन ट्रेनिंग, इट इज ऑन PCR एंड basically I have to learn from you. Primarily I was given 90 minutes, now it has come to 40 minutes, but then I'll postpone your lunch little more time. That's because otherwise it will be a mess, you know. If you are prepared for 40 minutes then it's fine, but if you are not prepared then इसका तो जरूरत नहीं ना, बट ये वाला ऑनलाइन का इसका तो आपको सुनाई दे रहा है ना बिना विदाउट दिस आई विल बी एबल टू हियर, डोंट वरी. With time to come my voice will again raise also, so don't worry. Now the question is PCR, now even the school people they are learning, you know, and after COVID everybody knows about PCR, at least they know what is RT-PCR, even means our mothers also they know CT values. किसी का कोविड आया तो पूछते हैं कितना CT वैल्यू आया, means that is what of late in 2021 के बाद 22 में तो लोगों को पता चल गया. So but then I was told to tell you about the PCR. So whatever I say, more than that you make it interactive, in between you can ask and I should learn from you more. And what happens usually, MSc means PhD has not gone to PhD, MSc they are still continuing, not got the degree. How many of you are here? So 1 2 3, right? Only 3 people are MSc. Of course MSc also means you know very well what is PCR, right? So who has done who has discovered PCR? Carrie Mullis, which year? Now if you search, no, you will get somewhere 1983, somewhere it goes 1985, right? So nobody knows, probably he has reported first time in 83, then the paper has come maybe in 85, whatever it is. And he was working in a company, know the company's name? Cetus Corporation, right? So, and Cetus Corporation is a wonderful company, you know, they have first time found the Taq polymerase also. You forget about my slides, they are for you, and whether I cover, skip, don't tell, whatever it is, keep with you. Some point of time if you take class somewhere it will be useful, because this is quite comprehensive. I tell in the class also, because see, if we go read slides then there is no point talking to you, right? So of course I have to, for getting the clue or the link, I have to go through this. So Cetus also discovered Taq polymerase, understood? So this is the person. This is in 1976 itself they actually discovered Taq polymerase, and they had patent for both. And afterwards of course Roche has purchased them. Roche is the company who goes for diagnostics, and most of our clinical diagnosis, best diagnosis is by Roche. Whether you buy a glucometer, whatever you AccuCheck mostly that is sold, that is by Roche, and otherwise also Roche is in the field of diagnostics, especially in the medicine. Of course in the plant science also it comes. So Cetus was holding them, then they have sold in a huge amount to them. So this is a beautiful means this is a wonderful company of that. You know the process, right? PCR सबको पता है, and I hope everybody knows a bit of Hindi, because here the Delhi culture is mixed language, you know, otherwise we don't feel much comfortable. We can talk but still we don't talk, means मिला जुला के बात करते हैं तो अच्छा लगता है. So in this process basically this is a age old, means I think it is in 2007 or 8. I have taken this from somewhere, and I was teaching this for the undergraduate when I was in Andhra. But then this holds true even today. You know, this is a nice slide because in one video they show everything. You have 3 steps, basically denaturation, which goes where your 2 strands will be separated, then goes to the annealing, and then it will be extension will be there. That's fine, this is all right. And from the same place itself I took this. If you are going for actually what is called 4th cycle, this is what is the status, and if you go to 34 cycle, you get 34 billion copies. Understood? So means from 1 copy. But when you put your template, that is not 1 copy. You put in quantity, maybe 5 nanogram, 10 nanogram, 15 nanogram, whatever it is, they will be having so many copies. Okay, so 1 copy itself gives you 34 billion in 35 cycle. So why do you keep so much? इतना हम कहाँ रखते हैं? Anything. So much, this quantity is huge actually. So why do we keep them? हम तो यूज करते हैं एक 0.2 ml ट्यूब, 200 माइक्रोलीटर. सो बोलो. Actually we don't get that many copies. Initially it works, and after some time, because of many constants, it gets slowed down. So that is why, like a bacterial growth curve, this is also having a growth curve. Initially it will be log phase, then it become like a linear phase, then it goes to the lag phase where it is saturated. Like if you do for 35 cycle or 100 cycle, you will get similar quantity of band because things are not there, right? So that's all it is. This is what is written there. You read if you get time, but what I am telling, that is what is written in nice language. That's all it is. Okay. So what are the components? What are the things required for that? Yeah, tag DNA polymerase will be required. Primers required. Template is required, and then buffer is required. So physical component, we need a machine, right? Can we do PCR without machine? So C can do. You can do. How do you do? Yes, water bath. So you need some sort of a incubator, right? But suppose I don't have a water bath, I have just one incubator or nothing is there. Can you do PCR? I don't have even a water bath also, but I want to do polymerase chain reaction. This is, I want to do that. Any possibility? No possibility. But the end of the class, at least you will know that yes, it is possible. And it is not only possible, it is being done. Especially in the medical science, there are many diagnoses is done which we call isothermal PCR. आप एक ट्यूब में सब कुछ डाल के छोड़ जाओ, next day you come and then run in a gel, you see whether the disease is there or not. So, बोलो? Yeah, LAMP PCR is one of the variant. It is there. LAMP PCR. See, मेरे पास जितना टाइम है ना, उसमें मैं खाली नाम ही टच कर सकता हूँ. I was just telling Dr. Anil also, usually when I was taking PCR class 2011 after coming, this used to be 3 hours class, means 3 classes. So that has been pulled, but then doesn't matter. If your names are there and a little bit in the slides are there, she has told correct, LAMP PCR, but आपने बोला नहीं कि नहीं हम बिना machine का भी कर सकते हैं. मेरे पास कुछ भी नहीं है. You don't have anything for LAMP PCR, you don't have to do anything. You just put all the reagents, ट्यूब को तुम्हारा टेबल पे छोड़ के चले जाओ, next day you can come and then run in the gel, you will see that your fragment has come, right? And it's not only the LAMP PCR, of course LAMP PCR is one of the variants, but you get a few. So we'll come at the end of this, okay? So again, I'll skip these things. This is denaturation, and then denaturation first when you start, no, you do for a little longer time because शुरू ही नहीं हुआ. Understood? So you do that. And then after that it comes to annealing, and after that extension, and how much is the extension time, that all depends upon how much big is your length. छोटा के लिए आपको unnecessary 1 minute 2 minutes देने का कोई ज़रूरत नहीं है ना. This some problem ना, यहाँ टच वच करने से कुछ हो जाता है. नहीं मैं अभी क्या करूँ, मैं तो कुछ टच नहीं किया. I am not anything. Is tick is coming. So many ticks are coming. This is चलो हो गया चला गया. That's no problem. Oh, I think this has to be done. Okay. So this you usually do that, right? You decide how much time has to be given depending upon how much is the length. Like in case of a qPCR, usually you have seen that the PCR is over by one and a half hours itself. But when you go for a normal PCR, it takes a long time, right? It's a longer time because usually your stretches are big. Many of the cases, even 4-5 kb fragment also we amplify. So naturally 4-5 kb will take some time, no? Somewhere in the slide is there. Usually TAC polymerase is having a capacity. How much usually, any idea?
500 base pair per 30 seconds.
Good, very good. यही लिखा है 1000 base pair per minute. Actually 60 base pair per seconds they say, but on an average if you take, then it will be around that only. It's 1000 base pair per minute. So based on that, these are all thumb rules for doing that. But nowadays we don't use any rules. Why? Because the tools are already there. You sit in the computer. If I ask any one of you multiply 375 into 220, no one will do multiplication. That's because you will just open the earlier calculator. Now you will open the phone and do this, right? So same way, even if I say so many things, none of you are going to do. I know that because there are software, there are tools. They use different algorithms. Like depending upon when you design a primer, you will be there is a formula, you know. And based on that, how much is G, how much is C, how much is AT, based on that, they calculate. But then they have come out with a different other formulas also, and we really don't bother nowadays much because you put, there are many good softwares are there, and free softwares are giving. Why they give free software? Because you will be buying primers for them. So they give it. Otherwise also there are paid software where you can do. Anybody has used mineral oil in case of PCR? Why you used? But मैंने किया वो सब के लिए नहीं. That was because the PCR was not discovered the way it is now. Understood? So after one cycle, when you are denaturing at 94 degree or whatever, the entire things will get evaporated unless and until on the top there is a heating subject. That's what it is telling Peltier. Peltier physics ka hai, but then what it does is your hot bonnet happens. Means on the bonnet also it is extremely hot. But now I'll give you a clue. Suppose your PCR machine, you are standing, you are there in a lab where you have nearby 100 kilometers there is no PCR. You had one that is gone, and this one, the hot bonnet is not working. That happens many of the times, and they will charge at least 1 lakh rupees to replace this the top one. Understood? Because it is hot, that's why it does not get evaporated and stay. नीचे का block काम कर रहा है. That block is actually that block is working, and your everything is working fine in the block, but top hot bonnet is not working, and because of that your things will get evaporated. When the first PCR came, first means at least for first quite a few years, so that time after putting all the reagents, we used to put 15 microliter 25 microliter में हम करते थे. So 15 microliter of mineral oil, so that will not evaporate at 94, so everything gets restricted there. So what I am telling is, in case your hot bonnet is not working, don't stop your work. Mineral oil is very easily available in any of the companies. You can do an exactly same result you will get. An interesting thing is, you don't worry much when you are loading in the gel. When you are loading in the gel, oil will automatically float and come out, you know. So it is not going to go and hamper in any way. And entire my PhD is with mineral oil. Of course, that was the first PCR in IRI in 1993-94. LEP Prem, one company was there. So in the chemicals component, you have already told all this, right? There are few things important in that. Nowadays again that is not important, because you get a master mix. Most of the cases you go, you don't bother about what is there in the master mix. But in spite of that, I'll tell you, in the buffer, they have potassium, which is very important. When you want smaller fragments to amplify among many bigger fragments, your potassium concentration goes, KCl concentration goes little higher. Means if you are using that manually, वो हम करते नहीं है, इसलिए हम trouble shoot भी नहीं कर सकते हैं. Many of the cases we don't know what component is doing what. That's why everything is pulled together. एक बार नहीं हुआ तो even my students I am telling, if I ask if it is not coming, क्या करोगे? Sir, repeat करते हैं. Means it is understood that if you repeat that it will come. Then nobody thinks why it did not come in the first time, why I repeat and it will come again. It's not necessarily that it will come. वो आने का जरूरी नहीं है. And हम कोई नहीं देखते हैं. Magnesium is almost like tack. If you add more, non-specific binding becomes more. We reduce when your primer is very very specific and you don't want any non-specific amplification, reduce the magnesium concentration. But then none of you are going to individually do. I did, because that time वो PCR evolve हो रहा है 93 94 में ना, 93 में तो उनको Nobel Prize ही मिला. So this is the thing, you know. Now primer is one of the important thing. Usually 20 base pair का हम करके देते हैं, and that also they do by the software. But then you are seeing that your non-specific bands are coming more. Very simple is you increase the length, because one nucleotide you are adding, your specificity is increased by 1/4, right? So दो दे देंगे तो means उसमें से बहुत सारे कुछ छाट जाएगा, right? So this is what you can do. This is what I am telling, that 4 into G plus C into 2 into A plus T, that is how they used to calculate, but there are other formulas by other companies also. Okay, so and then there are standard thumb rules by which even if you don't have a software you can calculate, means you can design your primer, which we used to do because that time computer itself was not there. So primer itself has to be designed by us. So this is what different algorithm by different companies they put it. And if you go by different companies things, you will see that TM values are not matching, because one thing is TM value depends upon your salt concentration also. आपको किस में दे रहे हैं? When they are giving, no, they say that there are different category of primer giving. They will say HPLC prepared, then they will say they are desalted, right? Dialyzed and desalted ones, because they will be having salt when they are preparing, and after that if you are not, when you are synthesizing you have to use something, but then when you are going to use you don't want any one of them. So it has to be basically desalted and given there. So DNTPs, you know, it is very much required. Since DNTPs are required, similar kind of things we do in case of sequencing also. So I'll come to the application part fast. In that, in case of sequencing we use, and chain termination, it's actually synthesis, no? So earlier we used to do just manually using a normal DNA polymerase. Understood? You use a primer that is means labeled with radioactivity, and after that what we do is we will be putting a normal DNA polymerase. So it will be joining one by one, and wherever there is a deoxy, means DD, ATP or GTP, there it will get stopped. The moment PCR has come, everything is stopped. Now we put everything in PCR, and not only in PCR, we don't use individually. There are, I think, 2-3 slides. I will skip them since I am telling them. So earlier used to do A is one tube, T is one tube, G. Once the fluorescent dye came, that time we put everything together, and then the fluorescence are detected with a photomultiplier tube. This is a boom actually, that photomultiplier tube, because of that qPCR is happening, your cell sorting is happening. Many of the equipments where just by that sensor they detect the fluorescence and then they say. So in case of sequencing, nobody nowhere uses manually, all the cases they use PCR. Not only your Sanger sequencing, but for your NGS also we use PCR. Understood? There will be a bridge PCR, means Abhi sir ne bol ke gaya, he is the master of all the kind of sequencing through NGS, high throughput sequencing. Wherever you go, there will be different sequencing, and every cases it has to be put inside the PCR. So you cannot think of a what is called any kind of molecular biology without application of PCR, and it is like if it is there with you, it is up to you to plan and think कि मैं इससे क्या कर सकता हूँ. आपके पास you have a sewing machine at home. Now you have learned making pant and shirt, but nobody is stopping you to do anything you want. You can make a design, you can make a handkerchief, you can make whatever kind of things as per your imagination, and that's what that fashion designers do, you know, and so much of money they get. So same thing, see, like how much ever difficult maths it is there, it is by addition, division, subtraction and multiplication. How much difficult it is, the combination of different things goes and you solve a very difficult problem. So PCR is such an instrument, it is up to your imagination कि मैं यहाँ पर एक म्यूटेशन करना चाहता हूँ तो कैसे करूँगा. I wanted to detect something, how we'll do that. Few of the things I'll tell within my time, but then it is you can just think of little help you can take from there and do. Okay, so TAC polymerase we have already told, but TAC though discovered in 1976, but then people thought why it is only aquaticus, Thermus aquaticus को छोड़ के भी हम तो और भी ढूंढ सकते हैं ना. They found, but then there is a list of how they are efficient and this, but still we are going by that. Of course there are lots of variants, understood. When you give for a sequencing, हमारा बच्चे लोग जो हैं, I want to know the exact sequence. See, you are doing a genome editing, you know. In genome editing, I want to edit one particular variety of rice. I am working with a N22. I want to edit a particular gene. So हो सकता है there is a SNP in that particular gene, right? हो सकता है because genotypic variation will be there. So before I think of designing guide RNA, I have to check the background, right? So what I have to do is I have to sequence that. Now when you go for sequencing, there can be a sequencing error also, because they also will sequence with TAC polymerase, right? When you are doing, what you will do, you will be having primer from both end of the gene or at least both end of the guide RNA. You will sequence, and that stretch you will be sending for sequencing, and if you have a machine you will go there and sequence. Believe that your machine is giving a very good sequencing, but your PCR may be wrong. They might have incorporated something wrong in that case. The stretch has come because of misincorporation. So that point of time there is an enzyme which they call high fidelity. They don't say exactly what it is because other company will start taking the bacteria and do. So it is like patented, but high fidelity in the sense they go by proofreading also. They have a proofreading kind of thing, means exonuclease activity also is there. So what high fidelity means, the error of incorporating a wrong nucleotide is almost nullified, understood? And that is extremely important when you really want to. It is not that you are getting a sequence and depositing in NCBI and writing in your name that I have sequenced. You are going to work on that sequence. Try to remember, you have a background. By mistake, you have not noticed that there is a mutation because of PCR or whatever it is, and then your PAM sequence you have designed according to that, and then after 2 years you are seeing nothing is getting edited. Understood? So there are tags different way. Magnesium I have already told, Kc I have already told, and you know this is how it goes. This the number of copies I was telling, no, 34 millions and all. This initially it will be slowly going, then suddenly it goes. This is what theoretically it goes in that way. But we don't have that much of space, we don't have that much of substance, we don't have that much of DNTP. So after some time it actually gets stopped, and that is what is that, you know. So we call it is like logarithmic. So what is log actually? Log में तो हम log 10 is how much? 1. And here it is that log and this log, what's the difference? You know, base. So here it is 2 base. What we are talking about, the log table, that is a 10 base. Understood? So it is 2 base. So in case of log, where we are talking about the pH, suppose pH1, right? If it is pH1 of a solution, you want to make it to 2. So how much you have to dilute? 10 times, exactly. So 1 molar is 0, 0.1 molar means 1 acid I am telling, 0.01 will be 2. So 10 times if you do, it becomes 1. But here 1 cycle is 2. So that's why it is log 2. Even in the case of bacterial growth curve also it works with a log 2 sort of a thing. So it goes. And it is extremely important when we are going for the qPCR. Understood? This log phase, lag phase, I am not bothered. Finally PCR हो गया, मेरा एक बैंड आ गया, बस मेरा जीन है, I am खुश. But then in qPCR you get completely wrong result if it is not understood and they are not calibrated in that way. Okay. So that is how I am coming to the qPCR. There also, you know, basically we actually quantify the number of copies of RNA. But हमारा जो PCR machine है उसमें RNA को amplify करने का कोई तरीका है नहीं. There is nothing. You have to make it to DNA, then only it will happen. So that is why it is first called reverse transcriptase. Okay. Most of the cases viruses actually does, so those enzymes has to be taken from the virus only. Whether you means whatever enzyme you take. So in case of your TAC, that comes from the bacteria, but for your reverse transcriptase enzyme, it has to be from the virus, and then the RT enzyme they sell it. So RNA will be made it to DNA, and then from there you will amplify. Now 1 copy of RNA will make 1 copy of DNA. So suppose I want to make a difference between a stress plant or and a normal plant by a qPCR of a gene, how many copies it is coming. So basically what you are measuring is how many number of copies of RNA are there, and usually we are always happy with relative, means इसके हिसाब से इसका कितना fold change हुआ. Why I am telling you, know, none of, I can guarantee none of our labs are calibrated. ठीक है, हम जो वे करते हैं ना 0.1 milligram or 10 milligram, means I am not very sure that that is 10 milligram. Understood? Even your nanodrop also is like this. Many people comes to my nanodrop, I don't believe my nanodrop, but many people come, बोला सर वहाँ पर रीडिंग ठीक नहीं आता है, हम आपके वहाँ means, but then still we are doing, we are publishing, even in our ICGB to everywhere, nobody calibrates all the equipments every year. Understood? But still why we do, because it is most of the cases our results are, our work is in comparison basis, means like how much, how many fold, how much percentage. Understood? So there it does not really matter. Now coming to this is reverse transcription, different enzyme they are talking about, and in this again you know very well, so I am not going into that, and this is how it goes. You have to have a oligo DT primer so that mRNA will be getting amplified, and then after that once the copy of the cDNA has come, then you are going to make it कि हमारा जो stress है particular gene has gone 10 folds, or understood how many folds it has gone into that. For that qPCR there are different kinds of primers are there. Okay, so this is one of that, like TACMAN probe they will put in the primer itself, they will put, you tell this is the primer, they will design and give, there will be a quencher, so chupchap rahega. The moment it goes and binds with the anil, and that time the fluorescence will come. Whether the extension is happening or not, it will happen. Why it will not happen? But the moment it binds, that fluorescence comes and it goes in that way. Okay. This is, I have manually done it while taking the class to make you understand where it is actually going. So see, this is the portion. Up to this you even cannot detect, because cybergreen otherwise also DNA में intercalate करेगा, means inside the DNA it will be there, but this is like a blank, this is like a blank. So this is made as a baseline. Now because of the amplification, how much of fluorescence is getting increased is getting counted, but up to this cycle, maybe 20 cycle or whatever cycle, you are not able to detect that at all. And that is how you in your qPCR graph you see like this. But the moment you come into this log phase, that means 2 will become 4, and this is what I made a comparison. See, one S1 is having this much copy and S2 to 2 samples, then basically it is double. After 8 cycle, 7 cycle, you are seeing that is 1 and 2, the unit of fluorescence. So 2 to 4, 4 is becoming 8, 8, 16. So after 15 cycle, 256 and 512, after that it is not actually getting proportional. 512 this is there, but after certain cycle you will not get proportional. So whatever your CT value, that has to be within that. You understand? Number, suppose one copy, when it's becoming 512 copy, when there is a 2 copy, it has to be 1024 copy. मतलब exactly double दिखाना है. When it goes to linear, then it will not be proportional. So your that CT value, what we are trying to find out, the number of cycle where this is in a log phase, that particular period is very, very important. But after that it goes in that later phase, no, it goes to the linear phase. See, it was double, but इसमें 2200 दिख रहा है, इससे 2400 दिख रहा है. So this cannot be counted, no. So your CT value cannot be taken when it has crossed the logarithmic phase and gone to the linear phase because of whatever constraints it is. Understood? One thing I forgot to tell you, suppose when there was real-time PCR was not there, we wanted to know that gene expression, whether it is increasing or decreasing. How do you know? How, what do we do in that? Normal PCR, right? It is a normal PCR. So you do 15 cycle, 20th cycle, 25 cycle, and then every cycle ke baad you just load that in the gel. Then you see the intensity. Exactly you will not get, but some idea you will get. See, in 20th cycle, in one sample I am getting a very dark band, and other one I am not getting the dark band. Understood? So that is how it goes. Now this is one thing all of you are doing. Melt curve you see. When you are doing a PCR or a qPCR, PCR में भी आता है. When you are doing, any one of you see the melt curve? Most of you don't see. You see because you are saying yes. Why do you see? Yeah, single amplification. मैं हमारा language में बोल देता हूँ. See, the primer has a non-specific binding, and because of that the fluorescence is getting increased, and you are feeling कि मेरा gene double हो गया, triple हो गया, whatever it is. But it is not that. It is basically binding in somewhere else and giving another band. Nothing will be seen in the gel, and even nothing will be seen in your qPCR machine because you are taking data. And what is happening is, if you even if you are running in a gel, if the size, and it will be the same size because your primers are fixed. 300 is your amplicon size. If it is binding in somewhere else and binding for some other gene also, you will get the same size, right? So if you are getting the same size, even if you run in a gel, you will not get 2 bands. So what's the way? हमने सर जेल में रन करके देख लिया, एक अच्छा सा मोटा बैंड आ रहा है, so it is single amplitude. It is not so. Melting is basically you are denaturing by heating. When you heat it, when you slowly heat, it gives a hyperchromicity. वो हमारा एक practical हुआ करता था तब. So you take the spectrophotometric reading at 260, you will be seeing that it is increasing. Okay. By that way, basically melting temperature, Cot value, all that you used to calculate earlier. Now it's not done. But then this is, you don't have to do anything. After PCR is over, you just ask your PCR machine, means in the real-time PCR software, that I want the melt curve. They give 2 types of curve. One is dissociation curve. This is this. This dissociation curve means you are actually, when with the temperature, when you are denaturing, that intercalating was more, so slowly and slowly the fluorescence will decrease. And if it is coming sharp this way, this actually is Alkas, my students thesis se main utha ke kal laya, and this is from the net. If you don't get this shows that this is a single means amplification. And this they see per unit of, per degree of change, how much of increase in your fluorescence is happening, and that is plotted in this way, and you will get a peak. What she says peak. If you see exactly same peak, that means it is your only one amplicon is coming. So your gene it is. But if it is, you are getting a dissociation curve of this, and you will definitely get 2. That means basically आपको size तो एक ही है, but then आपका melting curve में 2 peak दिख रहा है, means there are two different things got. So whatever values you are getting, they are not correct. So please see. It's nothing, it doesn't cost anything. Just you go and Google ki, because different software, no, different company software will be having a different option, so you can go by that. Now coming to this point, with that PCR will be over. I'll just try to take you to the application part a little bit after that, because that is what is we are supposed to. Now आपको एक PCR खरीदना है. Somebody is telling it is 6 lakhs rupees, somebody is telling 2.5 lakhs rupees, and you have money also. So which one will buy and why you will buy? So this is what is called social networking, asking their credibility, how much is the service, all these things you have to do. But I am talking in terms of a scientific as well as technical part. हम क्या देख के उसको चूज करेंगे कि ये अच्छा होगा तो इसको हम खरीदेंगे. For a normal PCR what and for a qPCR what? बोलो. नहीं, you need for many purposes. हमारा किसी भी तरह 5 लाख रुपया मैंने जुगाड़ किया and उस लैब में हमें सब कुछ उसमें करना है. I don't know tomorrow what I'll do. Understood? So what will be my preference? Usually what you see for a PCR. And पैसा बढ़ता क्यों है? Why the cost goes more? बोलो? Yeah? Tell. You are all coming, but then I want to hear one terminology. No, that's okay, but what I am telling is what you require. बोलो. रैंप ना. सो जिसका रैंप ज़्यादा है उसका प्राइस ज़्यादा है. व्हाट इज रैंप? या रैंप इज ए स्लोप. ये PCR आने के पहले तो रैंप मीन्स कैटवॉक होता था. मीन्स दैट इज व्हाट वी नो. अदरवाइज वी नो इन हॉस्पिटल. Even now because of Sugam Bharat, everywhere they have put a ramp. Basically it's a slope. So slope is having two axes, you know. One is X and one is Y. How much of raise per movement means per distance. So same thing per degree how much of the temperature, and maximum is better because आपको 94 आ गया. From 94 I have to come down next to again 50 or so. So if it is taking more time, I have to waste more time, and that is why they are पहले aluminium किया block, then they are telling silver block, they are telling gold block, some नाम दे देते, and then they cost more. Understood? So that is how it goes, and you will say that I will buy this because it will be doing fast. But can you imagine where I can go extremely high ramp? Maybe 5-6 is the ramp what we get. I am talking about 1000 ramp. That means per second it can even go up to 1000 degree. Just imagine 5 second करके कुछ भी बोलो. See, when the PCR was discovered, that was with the 3 water bath, that was having the extremely high RAM. Only thing is कितना जल्दी आप एक से दूसरे में change कर सकते हैं. See, it doesn't take time. Three water bath is there. You are putting in one, and तीन friends बैठा हुआ है, आप pass on कर रहे हो, या तो you yourself is doing in 3. So basically it takes at least for 30 seconds 40 seconds to come to one to another temperature. You take only 0.1 seconds or 1 seconds, right? And that is being used now. Okay, आप ज़्यादा दाम देख के 5 लाख 6 लाख में खरीदो. But in 2017 I saw in UK they are using a robotic hand. 3 water bath है, very precise water bath they are doing, and nowadays robotic hands are quite cheap, you know. You have made a program, 30 seconds you will be there, hold, put it like this, put it like this. So it takes only few seconds, you know. And so this is the one, basically similar kind, but this is like a what is called old fashioned. You get a very precise water bath, and in that you have put a crane type hand which will hold the plate and put it in one and another, and it goes, आपका 4 घंटा का काम 2 घंटा में हो जाता है. And this is a company, Isogene. This machine I have seen in Monsanto's, means like अभी Bayer का है, उनका जो machine है stacked 16 PCR at a time, you know, एक के बाद एक. ये साइट पे जाना, I could not, there is a video also with this, I could not give it to you. You just go to Isogene, they have given. इसके अंदर में ना इतना बड़ा machine है, इसके अंदर 3 water bath है, and they are so fast moving. See 30 seconds के बाद ना ऐसे ही, मतलब they don't have a robotic hand, but that plate is put there, that is just sifting. One water bath, second water bath, third water bath. So इतना बड़ा है, आपका तो एक प्लेट इतना ही बड़ा होगा ना, this much size. So it is going that. And they say the ramp is more than 1000. And it is obvious, no? See, आपको तो time ही नहीं लग रहा है. It is not in the same water bath, it is cooling and heating, it is 3. And in for their quality control lab, they have 3 this kind of machine which is stacked with 16. Now qPCR का एक चीज बोलना है, when you are going for a qPCR, if you can afford, this is available. If you can afford, this is there, you go by this. This is no replacement of this water bath of Carimulis. And if you are going for a qPCR, you will be getting 2 colors, 3 colors, 4 colors, 5 colors. What are those colors? And depending upon that, the qPCR price starts from 8 lakhs, 9 lakhs to 20 lakhs, 25 lakhs or so. So 7 colors I had when I came, Dr. Bansal's lab I was, and he has a machine which is having 7 colors. What are those colors? Any idea in qPCR? Yeah, 7 genes at a time. Basically 7 different kind of fluorescents they can detect. So every probe, suppose you are making a TACMAN probe with 7 different fluorescents, so they can very well find it out. Understood? But very interestingly, ये हो गया वो मशीन का 20 साल, अभी तक साइबर ग्रीन छोड़ के कोई यूज कुछ नहीं किया. Understood? We use cyber green and cyber green and cyber green. Then comes your point, what is the need? This is required, 7 color, when a routine diagnosis happens, you are able to detect 7 diseases of a person for a single blood sample in a AIMS they will be having, so that you have TACMAN probe से आप 7 probe बना के रख दिया, एक sample में कर दिया, एक साथ आप 7 disease को multiplexing करके बोल रहे हो. But then for you wanted to see gene expression, you are using only your what is called cyber green. Why do you want that? Single color का आता नहीं है, 2 colors का आता है, that comes around 8 to 9 lakhs. So this is what I wanted to say. This is useful things. When you go, don't go by 5 colors, 6 colors, 7 colors. You will not be doing, and it's very very difficult. आप एक बार के लिए TACMAN probe बनाना is also very difficult. So it goes by this. Now you see, usually what we do, we see the PCR product. This is the second part of that, where we do electrophoresis and see different size. Many times we differentiate based on the size. Then we take that size and give for sequencing. Different kinds of things we do in that. Now what for we use? Initially we all started using PCR for polymorphism, and specially जो तुम्हारा थोड़ा सा सीनियर लेवल है, all biotechnology labs started doing what is called RAPD. So they had a random primer, put it there, do that and see the variability. Basically for the sake of variability we do that. So based on present absent or different sizes, so you see the different polymorphism. And at the same time this technology, when today also it is being used, डॉक्टर अमोल से हमारा कुछ टिश्यू कल्चर प्रोटोकॉल स्टैंडर्डाइज करने के बाद वी डू द जेनेटिक फिडिलिटी, कि हमारा टिश्यू कल्चर के वजह से बिकॉज ऑफ दैट देयर इज नो वेरिएबिलिटी अमंग देम, सो वी यूज सम स्टैंडर्ड मार्कर्स एंड सी दैट देयर इज नथिंग इज हैपनिंग. एंड this is different kinds of things we do. That's what I mean to say. So this is what is the picture where it shows, yes, all the regenerated plants are same, but then you have to use PCR. Endless kind of thing, you know. Then you do classification of the organism, right? You have done the sequencing. If anybody is there from bacteriology or so, you will be doing ITS region, and then you will be comparing the sequence after doing the PCR, and you do that, right? This is one things. Many cases you do RFLP of the PCR product. So PCR usually if you are using for a RAPD, you say it is a dominant marker. So you wanted to make it a codominant, you just digest that, and then of course you put radio level also, and then you can actually get this kind of band, and then of course you can do the detection. Parentage for human being to plants we do for parentage detection. Then for cloning, usually you want to clone a gene. What you will do, first you will amplify, from there itself your PCR starts, and then you will clone, then again you will check with the PCR, so it goes. I am not going into that. Then coming into patient sample, because this picture was taken from a medicine book, but then this is what we do for disease detection in case of pathogens. Okay, so basically what it is, you have a what is called gene which is causing the disease. So if you have the primer, so if it is yes, then it is yes, if it is no, then it is no. So this is how it goes. And not only that, many cases you can identify when you don't have a qPCR, you can even identify, because qPCR के लिए आपको RNA से शुरू करते हैं ना, but then in this case gene expression we are talking, but in this case you can even see the load, ki how much it is there. And this is a typical example of mine which is done around 15 years back. It's been used, I don't know how frequent, but it is being used there. Ganoderma is a disease which you will be seeing this causing big plants falling, because they only eat the lignified tissues. तो एक छोटा छोटा प्लांट को नहीं खाते हैं, जब बड़ा हो गया, स्पेशली गुलमोहर, nice गुलमोहर plants, you will say next day it is falling because of this ganoderma. And early detection is extremely important. It's a soil-borne disease, so we take the root sample, and this is a bit of an improvisation. This was already published that they have specific ITS region from their primers has been reported. So what I did was, there are 2 sets of primer we took and we double check, and this is our sample. We used to go to the gardens, take the random sample for different root samples, but the problem with that is that root sample will be having plant DNA so much, your bacterial DNA will be having very less. So in that, this is basically fungus, that will be very less. So what do? We have to do something else. But then what I did that time, we put those root tissues in the petit plate and let the little mycelia comes. It's a matter of 7 days, means 5 days के बाद mycelia हम कर सकते हैं. So the day they give the root sample, we give the result after 7 days, because it becomes very difficult. But then in your PCR techniques, nested PCR is basically done for that purpose. You do 2 sets of primer, कि आप थोड़ा बड़ा कर लीजिए, and specific to that disease you do, and you actually amplify together. And with the nested PCR, such kind of situation, when you are going for a disease detection, your host DNA load is so high that you are not able to specifically do. You can go by this. This is little bit of written, you read there for DNA, nested PCR primer, how you actually are supposed to do. So this is another technique which is also related with that. This is a limiting primer with a higher melting temperature. So if you are having a higher melting temperature, that means only the specific primers will bind. So you are actually increasing your specificity. So that is how they are telling. First 20 cycles they do normal, but then they raise that melting temperature little, and they, most of the cases all the variants are same. As I am telling, no, it is like a stitching in a stitching machine, whether this way or that way will do, that is what it is. So they gave a name which is called late primer. Then comes to allele specific, which we quite frequently use. We design many of the cases for mapping also. If you have a SNP, and based on that you are telling, yes, this SNP is crucial. So at the last point, take the 3' one and design primer. You can actually get 2, even 1 will do. In one case you will get the band, another case you will not go. But if you are doing the primer with both the SNPs, then you get 2 types of bands. So this will be a double confirm of that. Okay, so this is called allele specific primer. Of course, DNA fingerprinting is used for many purposes. You wanted to identify the plants or whatever it is. Okay, asymmetric PCR, when I am talking the name, anything coming in mind, you forget, you read later. It's actually one direction, only one strand I will do. Understood? Most of the cases, in case of sequencing, we do that. Got it? We do in one direction. And when you do a probe for hybridization, this has to be a single stranded. Suppose you are doing a southern, you will be doing a one strand, no? So that cases we do single stranded amplification, right? And since we are talking about the sequencing, I put little bit, this I have already talked, so I am not talking on this. 4-5 slide I can very well skip. So at present they put together, and like as we are telling, no, they all the probes, all the DTP, everything you put together, amplify, and then it is done, it is detected by a photo multiplier tube. इस fluorescence का मतलब A है, green के मतलब G है, like this they go by that, but then it is started with your PCR. Now when we are talking about the sequencing, there are few slides which are on NGS. Next generation sequencing may overlap PCRs, and then this is ligation mediated PCR. I am not burdening you, coolly sit and do see that, but there are lots of variants of PCRs are used for NGS. So sequencing में तो use होता ही है, otherwise for different technology of sequencing they use, like that Polony PCR, bridge PCR, like solid phase PCR. Solid phase PCR में उसको actually solid के ऊपर ही PCR होते रहता है. Understood? So that is what there are few slides, as I am telling, no, it was for, and this is for bridge PCR. This is dial-out PCR, is a complex library of DNA molecules, is modified with unique flanking before massive parallel sequencing. The meaning is, what is happening is, it is at a time we are doing accurate DNA molecule for the gene synthesis. So आपको एक DNA library बना के जो flanking region है, that very specific to that, you just, for little detail I need 5 minutes time, but I don't have that. So if you want, you just go into dial-out PCR, you will get there. Now touchdown PCR nowadays nobody uses, because we are getting the PCR machine where you will be having gradient. तो आपको प्राइमर आने के बाद ही किसी को भी पूछिए, all of you who are doing, you will do, मैं सर आज प्राइमर को gradient लगा दिया. Why you put gradient? That's to know that exact temperature कहाँ हमारा amplification हो रहा है. जब वो सब था नहीं, तब के लिए touchdown था. They used to every two cycle they used to decrease 1 degree. तो उसमें से आपको 4-fold increase हो गया, ना, 4-fold amplification हुआ. So there you get your actually amplification. You are not knowing exactly what, but based on your TM and annealing temperature, you do a touchdown PCR. कोई ना कोई temperature में आपको enrich होगा, and that will give you more. Colony PCR everybody knows. If you have cloned a gene, you don't have to do anything, directly with the colony you go for that. This I will take 2 minutes because this is extremely important. Inverse PCR, what is that? What is inverse PCR? Why the name inverse? Bolo? Yeah, tell. See, PCR may, if it is, that's why I was asking for the board. Suppose you have a stretch, you have this one left primer, right primer, it is going there. Okay. Inverse is other way. One is going this way, another is going this way. But if it goes this way and this way, how you will get a product? Circular DNA. तो कब करेंगे हम? When you use it, you are perfectly okay. If you have a circle, this is my target. I don't want this target. I want outside portion. So when do you use that? Anything? I want to know, basically this is for knowing the flanking region. Understood? किसी का भी हो, आपका जो target है उसका बाहर वाला portion को हमें जानना है. Because of that बाहर वाला portion, take that outside and make a circular. Typically this is used, even today this is used, and most of the companies make a kit used for transgenic event detection. Your gene got incorporated in one place of the genome, right? Now where it has got incorporated, how do you know? Understood? If you do PCR, one direction go, you will not be able to see. So what you do, you cut with a restriction enzyme, then you ligate them, so they will become circular. So you get the target where it got integrated, followed by a circulation, then you do a PCR, and that outside portion you sequence, then you will be knowing what is two sides flanking region, and that is very very important. Understood? ये हमारा Monsanto ka BT cotton is, they say that is our event and sort of things, no? So for knowing, or whenever you are going for your transgenic, that is extremely important to know, this is what it is, you know. You first linearize, then circularize, then you make that portion, and then you can give for sequencing this portion. So one end is one flanking and another end is another flanking. So this also you can do, methylation specific, you can do. See, there are enzymes which we call isosisomers. Some are methyl sensitive, some are not. So you can cut with that. After cutting, you just go for amplification. MSP and HAPA is typically the example when we use even for methylation detection. So by cut with the methylation sensitive enzyme, and then you go for amplification, or I will not know. So this you have already heard of. I am not going. Most of you know tail PCR, right? So tail PCR is nothing but you have to actually find out what is the other end of this. Okay. So they designed some specific primer, and there are some hand tips also has been given for that. Other one also you are quite familiar, that is RACE. Tail PCR, RACE PCR, usually we don't think much because they give a kit and give the instruction manual, readily we do. Little bit you see this afterwards, I mean whenever you feel, and then you can actually go by this. And this is called differentially expressed PCR. See, usually before the transcriptome analysis, we used to do subtractive hybridization. Understood? Subtractive hybridization means you take the RNA of one sample, RNA of other sample, then you hybridize them and see which are hybridizing. Those which are hybridizing, they are basically common. The rest of the things, whatever has come, those you make a library, cDNA library, and after that one by one you sequence and say these are things. The moment transcriptome came, that got stopped because usme bahut time lagta tha, and this is a substitute of this. Of course, this is what is for creating a mutation and detecting a mutation. So it's very easy in the sense you wanted to create a mutation in a gene, especially in case of a bacteria or a circular. So what you do, where you want to create, there, that place, you have a matching portion of your primer. Other than the matching portion, in the flanking region, you put a different nucleotide what you wanted to make. So in case of a site-directed mutagenesis, we usually specifically incorporate that, and after 2 cycles, that will get incorporated and you will get that. If you actually follow this picture itself, you will know. And in this case, basically, it is done with a restriction site has been incorporated in the primer. So you get a restriction site, so then you can cut it, and then by that way you can detect that mutation has happened on it. This is also important. Okay, at least you know. See, how do you know the copy number of your DNA? Many of the cases it is required, especially when it is transgenic. You have to know the copy number. Now, how do you know? How you detect the copy number? Transgenic में हम सदन करते हैं. Usually we do the southern, seeing the number of bands, we say this many copies and all. But there are reports, and they are using also, though even qPCR, if you are going to a bigger journal, they don't accept that, you have to do southern. But this is what is done. Mostly you do a qPCR of a DNA, and then you have a standard. In this case, this is your standard, and if it is reduced, or if it is, this is for a disease basically, it's for a medical, but then in your case, if it is a standard 1 copy, if you have, along with that, if it is becoming 2 copy or 3 copy, the fluorescence will be increasing, and this is calculated by this thing, you know, the software. Now this is called co-amplification at lower denaturation temperature PCR. You read coolly, the people who are going for doing genome editing, this is going to be one of the things where you can detect the mutation. Understood? The name is given called co-amplification at a lower denaturation temperature. So they actually, what happens is, if you have a mutation, when it is a when the mutation has happened in all the chromosomes, you don't have any problem. But when there is a mixture that some has happened and some has not, it did not happen, then if you give for sequencing after amplification, you will get a jumbling. That is what we are keep on getting that. Understood? Because all the copies has not got. So you have a mixture. You have a mutated one as well as you have a normal one. So in that case, they do a low temperature. That is because where there is a mismatch, understood, there it will be a loop. So because of that loop, their melting temperature will reduce. Because वो तो अलग हो ही गया ना. Means basically denaturation won't have to go up to that. So lower denaturation temperature करके they have done in case of many of the diseases in tumor cells. Basically in case of a cancer, different variants happens. So they do that. We can very much, actually most of the techniques are first done in medicine. So you can very well do that in case of your gene edited, where there is a jumbling is coming. You don't have to do anything. You just reduce this. And they have given the beautiful protocol in that. Now this you might have heard of. I'll just do, one is emulsion PCR, another is droplet, digital droplet PCR. What they do is, they have a emulsifying machine, and each droplet is supposed to have one copy of your template, and then they multiply that. That is what is emulsion PCR. Little variation of that, the Biorad is having the patent, and then they say that is called droplet digital PCR. And here what they make is each droplet, some will be having 2 copies, some will be having 3 copies. I have written nicely. If you need, you just read that. Because of paucity of time, I am not going into that. But at the end, they use, their software use a formula of that Poisson's statistics. Poisson's coefficients ke basis pe they say ki how much of your gene is getting expressed in that. Understood? So this is from Biorad itself.
Hello,
this is
sir.
a PCR for
Audio is not coming, sir, in the Zoom
PCR,
meeting, sir.
and this is for droplet PCR. Here the precision, they say it is much more than the real time PCR. Means like when your sensitivity also, when you have very less difference.
Low battery, please charge.
So you can put that. This must have been talked by our Soham and this, in case of, this is our lab's work, so I'll just keep with that. You have like tRNA, guide RNA, scaffold. tRNA, guide RNA, scaffold. We initially used to go by level 0. So there you have to have a overhang, and probably Archana and Soham, they have taken the class of that, so I'll not talk because I was just going through that. This is the same thing. We create a overhang, and after that we do the PCR, and this is by the golden gate. Means like you are actually cutting, at the same time you are ligating, and then after that you do the PCR, and we find out that this is each level 0. Okay, after each level 0, we connect all the level 0 vectors along with one of the promoter as well as one of the linker at the end linker, and then we make, this is all 4 guide RNA. We have actually up to 6 guide RNAs, and they are successfully done there. And after that, when we amplify, there's a PCR, there is a little problem. I don't know whether Soham has told. See, what happens is, we'll be having the primer from tRNA and here from the last guide RNA. We get different combination, and most of the cases we get 2 guide RNA, 4 guide RNA, 1 guide RNA, 2 guide RNA, 3 guide RNA, all different kinds of band we get. But we know if it is 6 guide RNA we are looking for, which will be the band size. So we elude that, and then we again clone into the level 2 vector, which is this. This is we used in case of a rice for PRGV32, and then basically it goes. And I'm not going into detail because, and this is the steps of that. Okay. Now, of course, finally again you have to go for the sequencing, which I told that better you do that lower denaturing temperature. I have also not used, but then I am going to use that. But otherwise, if you have to amplify that guide RNA portion from your edited, and then finally you have to go with the sequence. I'll take another 5 minutes. I know you are hungry and it is late. This is the one which I am trying to talk about, that we really don't need a machine. He has already told about LAMP. Other than lamp, I'll go directly to the, okay. This is one of the variants where we combine the immunotechnique as well as, this is slides from my immunology class. So you actually do two things. You do, here it is basically immunocapture PCR we say. So first the virus is captured, you wanted to detect whether my virus is present or not. So that virus code protein specific antibody you do, and in a ELISA plate you take that. After that you rupture them and do a qPCR. So it will double check, you know. First you are only arresting your viruses based on your monoclonal antibody, whatever antibody it is, through in the ELISA plate, and then you can actually do this. Okay. पता नहीं क्या है, क्या करूँ इसको, unmute, mute जो भी है. So this is go by this. You take the samples, then you capture the virus, and then you do a real time PCR. आगे पीछे कुछ जा नहीं रहा है. Right? So that's what it is. Now coming to ligase chain PCR, this is what when I was doing 1994-95, this became a big thing. Nowadays nobody uses. They just get two primers, and they use, instead of a Taq polymerase, they use a ligase enzyme. Understood? Denaturation has to be there. So every cycle, if they join, that means you say the band is there. I'll show you the picture, you will understand. This is called LCR. So this is one primer, this is one primer, and in between there's a ligation will be there. So you get a bigger fragment. Okay. So that is how you get. Now coming to this, I'll tell you, I'll come directly to that. This is called NASBA, or like transcripted mediated amplification of amplification, or nucleic acid sequence amplification. In that they use quite a few enzymes, but if I come to the picture, you will know there is no PCR, nothing is needed. Everything you put in a tube and you can get this. This is a RNA target. We are actually detecting the RNA. Then you use a reverse transcriptase. Understood? And then this you use from this you use what is called RNase H, because reverse transcripted you will give the cDNA here. So this RNA is getting cut with RNase H, and after that this cycle you polymerize this. So this will be double stranded, and then you use T7 polymerase, which will be producing a RNA. So you are actually, from the RNA you are making so many copies of RNA, and this arrow is wrong actually. This should go to here. So it will be keep on going. You don't have to denature at all. Understood? It is reverse transcripted will work at 37 degrees. So 37 degree में ये RNA जो है उससे आपको double stranded means hybrid आएगा, cDNA plus RNA. This will be cleaved, and then again with the DNA polymerase it will be two strands, and from there T7 polymerase again RNA will be coming. So multiple copies will come. Next day morning you come and put it in the gel, you will see a band has come. So that means that virus is present in the blood or in your sample. This is the another one which is called strand displacement amplification. In case of this, here what happens is we incorporate one restriction enzyme which cuts in one strand. That is because this is already methylated one, only this primer we use. Understood? So what happens is this is what this primer you are putting there, and this is the site which it cuts, which this is hing 2 which cuts here. So first time you have, this was not there at all, so you have incorporated this. The other strand has gone and filled this, but then since it will cut in this place in one strand, it will be cutting in this. Now the moment it cuts, this is having a 3' OH, that 3' OH will push and this will peel off. So continuously the strand will be displaced and displaced and displaced, and finally it is no denaturation at all. So you are putting everything in one tube and going, and next day morning you come and see that there is a band, means when you run in that. Okay, so this is in one direction I am talking. Same thing can be done. You can put another enzyme, another primer in this and do that. So that is what it is done. Now helicase, you know what it does? Unwind. So ये denaturation के जगह आप helicase दे दो तो भी वो ये एक ही काम करेगा. So you are actually putting the polymerase and helicase together and put in your DNA and go home. Next day morning you will be seeing that your band has come. Understood? Because आपको तो स्टैंड को अलग करना है ना. So when you are going to, once it is done, then they will be doing that. This is the last one, your lamp. Understood? So this is loop mediated. Basically they will be putting a primer with a overhang and they will form a loop, and in that loop every time there will be halfway this. So what I told you, that stand displacement, no, if you just read it little bit you will understand that, because as I am telling, I just wanted to introduce with the terminology, so at least you go into that and whatever is required you can try. So because of this stands displacement, both side this will be keep on amplifying. So आपको कुछ नहीं करना है, everything you put in one, and it is quite standard. This kit is sold by न्यू इंग्लैंड NEB, न्यू इंग्लैंड बायोलैब का आप साइट पे जाओ, लूप दे दो, वो पूरा सारे रिएजेंट आपको दे देंगे, आपका जो जीन है उसमें सारे डाल के आप घर चले जाओ, नेक्स्ट डे मॉर्निंग यू विल बी एबल टू सी दैट, नो पीसीआर मशीन रिक्वायर्ड. So there are quite a few things. This is oil palm gardens photographed by me, but then that's all it is, and got little late. You can ask question now, but it is difficult to ask now, but any please. Which one? You use normal polymerase. Normal polymerase means not TAC. DNA polymerase हम use. I think I have one slide fast to skip. See, this amplification is done much before PCR is discovered. You know, we used to do southern hybridization. उस time तो आपको करना ही है ना. What we used to do, you know, we'll be having the DNA, put it in water bath. It will be single stranded. Immediately after water bath, you put it in ice. Okay, so that it does not get annealed. Then after that, in the ice, we use a random primer. कोई ना कोई bind हो जाएगा. Bollinger का kit आता था. Now also it comes. So you will be putting a random primer and a DNA polymerase. Any organism का DNA polymerase. You don't have to go for TAC and Thermus aquaticus and all. So basically that is much cheaper also and easily available also wherever you get. So normal DNA polymerase is not that which is heat stable. Understood? This is not because of your TAC polymerase. It is because you don't require a machine. Nowadays, since it becomes cheaper, nobody bothers about that. But some point of time, it was unaffordable with the income and with the money we had. And moreover, in chota diagnostic lab, if they buy the kit, they don't have to buy the machine at all. So as I am telling that you can any point of time you can ask through mail or WhatsApp or whatever if you have any doubt. But I know, I am very sure you are doing a different type of PCR, many of you in a different context. Everything cannot be covered, and it is every day somebody is coming out with a new improvisation of the PCR and giving a different name, and it is up to you. It is up to you. ये जो आपका नया एड जीन में वेक्टर मिल रहा है, they are also all used by PCR by including. आपको थोड़ा सा कोडन मॉडिफिकेशन करना है. Codon modification is simple. Just use the PCR, like as I am telling, site directed mutations. You just change one particular nucleotide so that your amino acid is not getting changed, but the codon is getting changed. तो फिर आपका पेटेंट छूट जाता है. That's what they say कि आपका पेटेंट में वो जीन है तो उस नहीं. So that's all, Dr. Anil, from my side. Thank you so much.
So we would like to thank you, sir.
Yeah. Thank you so much. Thank you. हाँ, इसको चलो खत्म हो गया. शाम को फिर बाय बाय.